honor on today's symposium. Uh, Other dignitaries, distinguished invitees, Dr. Professor Anupam Verma, who is advisor on ASU and ex-national professor ICR, Professor B.L. Jalali, former director of research HAU Hisar, Dr. B.N. Chakravarti, former professor and head in North Bengal University, and Dr. R.K. Jain, I think he has not yet joined, but he also will be joining soon. Dr. S.C. Dubey, who has joined from NBPGR, head quarantine, and other distinguished invitees, Dr. Usha Sharma, who is Emeritus Scientist in the Division of Plant Pathology, other distinguished invitees, all participants, scientific staff, other staff, ladies and gentlemen. It's my pleasure and proud privilege to extend a very warm welcome to all of you on the occasion of one-day symposium being organized by Delhi's own chapter of Indian Phytopathological Society through virtual mode on modern trends in systematics and bioprospecting of fungi. This topic is very well chosen in today's context that we are having very depletion of taxonomists, be it fungal taxonomist or microbial taxonomist or plant taxonomist. So, and also there is so much of wealth in fungal collection and the gene bank which we have in our fungi, and so much to do in exploring the biodiversity in fungi. And if you look into the Indian context, more than 1/3 of fungal biodiversity exists in India, which is still unexplored. So in this context, the topic of this today's symposium is very apt, and with the eminent speakers and all distinguished legendaries in the field of plant pathology and fungal taxonomy, I'm very sure that today we will have a very good deliberations throughout the day. With these brief introduction, I warmly welcome Dr. P.K. Chakravarti, who has just joined. And as I mentioned earlier, that our director has to join another meeting, which is Extension Council meeting, which is to be held at 10 o'clock today. So we would like, sir, I'm so grateful to you. In spite of so much of busy schedule of yours, you could make it a point to join this symposium inaugural session and give us a lot of inputs and ideas so that we can improve in our societal deliberations, and also in taking phytopathological society deliberations, and as such plant pathology at the national level and international level to a new heights. Dr. Manoharachari, I profusely and warm-heartedly welcome you, that you are such a legendary figure in fungal taxonomy and other aspects. And in spite of your recent recovery from COVID-19, sir, we are so delighted and so happy that you could join, and your blessings are always required to all of us, sir. Thank you so much for joining, and welcome once again. I would like to welcome Dr. P.K. Chakravarti, very dynamic leader. He is the president of Indian Phytopathological Society. At the same time, sir, every time whenever we hear you, we get new ideas, and we are so thankful for accepting our invitation and gracing the occasion. I extend very warm and wholehearted welcome to Dr. B.L. Jalali, sir. Sir, we are so happy to see you through this Zoom meeting, although we cannot meet personally, but we are very happy, delighted, and a very warm welcome to you, sir. Equally, I'm so delighted and happy to see Madam Usha Sharma. She had been there in the division for many years, and ma'am, your guidance, your support, and so much of your guidance in looking into the fungal diversity, especially in terms of mycotoxigenic fungi and also aflatoxin diagnostics, especially on Aspergillus parasiticus and its pathogenesis. So, ma'am, welcome to you, ma'am, and we will be really happy to get your inputs for our future research programs. Warm welcome to Professor Anupam Verma, sir. Sir, you are always a guiding force, and always so much we get motivation with your presence only. So what to talk of your good words and so much of guidance in our future programs. So, sir, I welcome you all with very kind and warm heart. All the IPS official secretary IPS, zonal president Pramila, zonal councillor Deeba, and other ICR officials, Dr. Malkhan, Dr. Kalyan, and all IPS invitees and participants and members of Indian Phytopathological Society. I think not only Delhi chapter members, but other members also have been invited, and they may join during the deliberations. So I welcome each one of you. When I was going through the program, I was very happy that there are a lot of posters, e-posters are there, and there are many entries in the Narsimhan Award contest. It was really very, very delightful moment to see that there are so many people who are enthusiastically participating in all the activities during the whole day symposium. So I welcome all the participants, all those who have joined for the various contests and also for the posters. So with these words, once again, I welcome each one of you and warmly welcome all legendary figures and stalwarts in the field of plant pathology, and also my warm welcome on my own behalf and on behalf of Division of Plant Pathology, and as ex officio member of Indian Phytopathological Society, I welcome once again each one of you. I won't take more time because there are other deliberations to be taken up in this inaugural session. So these words, I once again welcome each one of you, and thank you very much for accepting our invitation. Please, Amrita, you can proceed further.
Thank you, Madam, for the welcoming remarks. Now I request Dr. Robin Gogoi, Secretary, Indian Phytopathological Society, to kindly enlighten about IPS activities.
Yes. Good morning, one and all. Honorable Director, Sir, Director IRI, Dr. A.K. Singh, and Dr. P.K. Chakraborty is President, Indian Phytopathological Society, and most respected senior members. So I started from Dr. Anupam Verma, Dr. B.L. Jalarli, Dr. Usha Sharma Madam, Dr. Manohara Sari. So, and all the members of Indian Phytopathological Society and my colleagues. So in a brief, just I want to mention that about the Indian Phytopathological Society. All of you know, so it was established by Dr. Balachandra Bhavani Shankar Munkur in the year 1947, and the society was registered after 2 years in 1949. And to brief, so the society is considered or regarded as the third largest society in the field of plant pathology, having almost 2,500 members. And so far, because this society is having 8 zones, and so far 2 zones have completed their this zonal symposia. First in the 1st and 2nd December from Southern Zone, and 7th and 8th December from Northeastern Zone. And today we are assembling in this symposia, one-day symposium, this is organized by the Delhi chapter. So, and 2 more things I want to highlight here. So we have developed our website, Indian Phytopathological Society website, actually updated. And from this year, we have started the online mode of conducting the election, Indian Phytopathological Society election. So this online portal already started from 7th of December. And within 2 or 3 days, we are going to open up the application receiving through online mode for different awards, because the society is having total 17 awards. The first award was instituted in the year 1962 in the name of Dr. Munkur Memorial Award. And last year also 3 awards were added here. So, and for the students and also researchers, young scientists, so 2 important these awards are there. One is Professor M.J. Narasimhan Academic Award, that is contest basis, and the society is publishing the journal under the banner now, for this is the 3rd year, under the banner of Springer, this, and then Indian Phytopath News, one newsletter also we have started for, this is the 2nd year running. So where we used to publish first-hand information. And this IPS travel grant, American Phytopathology Society, so in collaboration with our IPS, so one contest, IPS travel grant also started in for award. This is awarded to the PhD scholars only, not the in-service candidate, and also not for the MSc student. This is strictly for the PhD students. So with these few words, I would like to welcome all of you to this Delhi Zone chapter on behalf of this Indian Phytopathology Society. Thank you. Amrita.
Thank you, sir, for sharing the various activities of IPS which aims to promote research and development in the field of plant pathology. May I now request Dr. T. Pramila Devi, President, IPS Delhi Zone and Organizing Secretary, Principal Scientist, Division of Plant Pathology, IRI, to give the details of the technical program of today's program.
Thank you, Dr. Amrita. I would like to extend once again a warm welcome to you, all of you, sir. And I thank you all for taking time out of your busy schedule and joining us for this event. And here, I'll show you some more. Are you able to see my slides?
No, ma'am.
No?
Sharing.
Not yet. Share the screen.
So in that case,
disabled to nahi hai?
Are not able to see the okay, and then I will read out. And here we have the program is modern trends in systematics and bioprospecting of fungi. Under this, we have 5 different themes, sir. And we have 4 technical sessions. And technical session 1 is consists of a presidential address and award lecture. It is chaired by Dr. Reshmi Agarwal and co-chaired by Dr. Dinesh C, Division of Plant Pathology, IRI. And both these lectures are for a duration of 30 minutes. And the presidential address will be given by me, and Dr. M.K. Patel Young Scientist Award will be given by Dr. G. Prakash, Division of Plant Pathology, and followed by technical session 2, that is keynote 2 keynote lectures it has. And one is given in the area of bioprospecting of fungi, and another one is in biosystematics by Dr. C. Manohar Achari, and the other one is will be given by Dr. Shilpi Sharma from IIT Delhi. And then we'll have a lunch break up to 2 o'clock. And then we'll join again at 2 o'clock for concurrent session 1, consists of e-poster presentations. Here we have 21 presentations. And this session will be chaired by Dr. A. Kumar and co-chaired by Dr. T.K. Bagh. And here we have another session, that is concurrent session 2. Here we have the contest, like award contest, the MJ Narasimhan Merit Academic Award contest and APS Travel Award contest. And this session will be chaired by Dr. V.K. Baranwal and co-chaired by Dr. M.S. Yadav from NCIPM. And here for MJ Narasimhan Merit Award, we have 6 lectures and 1 APS Travel Award lecture by the PhD student, and followed by the vote of the thanks. Thank you, sir.
Thank you, madam. Now I take the privilege in inviting the chief guest of today's function, Honorable Director, IRI New Delhi, Dr. A.K. Singh, to address the gathering with his valuable suggestions.
Thank you, Amrita. Very good morning to each one of you. Dr. P.K. Chakravarti, President, Indian Phytopathological Society and member ASRV. Our most esteemed teacher and mentor, Professor Anupam Verma. Professor Jalali, I have been in touch with him since 1985. You may not remember, there was a symposium like this in Banaras Hindu University. I was doing my master's in genetics with Dr. B.D. Singh, and I had a problem on Asclerotium rolfsi, and presented a paper there. I was thoroughly grilled by him. I still remember that particular day. But later on, again, I had an opportunity to get associated with him on a major project on NAIP, where we made efforts to bring in major genes for resistance to blast diseases in the background of one basmati variety, Pusa basmati 1, And this has paid a rich dividend. And during this implementation of this project, his guidance and critical suggestions was very, very useful. We know that European Union has put an embargo on import of basmati rice if it has a tricyclazole residue of 0.01 ppm. 0.01 ppm, mind it, 1 milligram in 100 kilogram of rice. And this is only possible when we have absolute genetic resistance. There is no way that you spray tricyclazole and yet you achieve this level. It's very difficult. And that is where this project, where we brought 7 major genes for blast resistance in basmati background, and one of the variety was released Pusa basmati 1637, which is now paving way for securing our export of 6,000, 7,000 crores to European Union of basmati rice. So thank you, sir, for your mentorship on that project, a very important project that was implemented. Professor Manorachari, I just saw his 2 books he showed, and we had, a lot of difficulty in understanding the taxonomy by Alexander. If I remember, in BSc Agriculture, Dr. D.C. Pant was my teacher. Because those books were written very, the language-wise, it was a little difficult, and having come from Hindi medium, after doing ISC Inter-Agriculture, High School Agriculture, and coming to BSc Agriculture, suddenly shifting to English medium, it was very difficult to understand Elogipolus and those books. But the kind of books now, that are available, and I think more and more people should come out with such publications. This is one thing I think I have a request from my colleagues in IRI. This is one front where I find that IRI has not really made that significant contribution in terms of coming out with the standard textbook for graduates and postgraduate students in any field, not only pathology or mycology. I'm saying overall in all subjects, barring few 2-3 cases, we have not really done that job. And I think this is high time that we should take up this responsibility with all the expertise that we have available, so that we educate the whole lot in the country. And this is very important, thing to do. Dr. Rasmi, the Dean and Joint Director of Education, Dr. Pramila Devi, President for Delhi Zone. I would like to extend a warm welcome to all the participants, to each one of you, to this virtual symposium on modern trends in systematics and bioprospecting of fungi. And this is so very important, as we know that the fungal kingdom is estimated to comprise of something like 2.2 to 3.8 million species, with only about 7% named and classified. Several approaches, including novel biochemical, physiological, and molecular techniques, have been utilized to improve the systematics of fungal taxa and estimates of their diversity. The multidisciplinary approaches are being used for resolving species and higher taxa of the fungi. However, even with all the benefits of new techniques, they are also providing unclear results and taxonomic instability. Taxonomists should be aware of these issues and should follow pragmatic approaches. In order to overcome these taxonomic challenges, cooperation and communication among mycologists worldwide are crucial for the study of fungal diversity. I was just going through some literature, and I could see that the future of fungal taxonomy is challenging. Traditional morphological studies are still very, very important, as species names are needed in plant pathology, quarantine, and most applied industrial aspects. Yet, morphology alone is inadequate and far too subjective. Therefore, taxonomists must incorporate modern techniques such as phylogenetics, genomics, metabolomics, and evolutionary approaches. Therefore, linking traditional taxonomy, applied research may well be a good way forward. The next and more difficult steps will be to develop community standards for sequence-based classification, single and multi-gene phylogenetics versus phylogenomics, defining higher ranks in the classification system, exploring the undiscovered based on environmental sequences. While we have a lot of data that comes from the environmental sequence analysis on the kind of diversity that exists in this particular taxa, making sense out of it is very, very challenging task. And that is what I think is a major issue that should be addressed during this deliberation. I am sure the focus will be given on some of these issues. Also, the evolution of these pathogen, is so fast that many times we find it very challenging. We were working on a disease in rice called, Bakane, and we have Vishnu Maya associated from plant pathology division. We identified some germplasm showing high degree of resistance to it. And then by the time we mapped the gene, we started marker-assisted backcross breeding, transferred to two major varieties, 1121 and 1509. And then it has taken 3-4 years time. And by the time we come to this stage with the product, we know that there are a number of other, fungal variants for Bakane that have come, which knock down these genes. So the additional deployment of genes becomes very, very, challenging, and simultaneous discovery of all possible variants of the genes that are available in the germplasm. Huge collection. For example, in rice, close to 1 lakh, plus accessions are available with zero. But unless and until massive efforts are put on screening and identifying novel sources, simultaneous deployment and their characterization, it would be very, very important. Like, I was telling of the BLAST program. Now we have in the background of one basmati variety, we have 1 gene, 2 gene, 3 gene, 4 gene, 5 gene, 6 gene, up to 7 gene pyramids, all major genes. And these pyramids are available in store. As and when required, they can be immediately deployed. Now that kind of efforts is required in other cases as well. And Bakane is one such challenging problem, fast evolution happening. So is happening in other pathogen also. So these are, and that's what I think there is a need for both the pathologists, the breeders and geneticists to come together, work together to solve the problem. We talk of U triangle in Bresca evolution. There is similar triangle in, the host pathogen and environment in case of plant and pathogen interactions. So all these three corners of the triangles have to work together to make sense out of it. I wish this virtual symposium a great success, and thank you very much for giving me this opportunity to share a few thoughts of mine with you. Thank you very much.
Thank you, sir, for your motivating speech and influential messages. May I now request our guest of honor, Dr. P.K. Chakrabarty, President IPS and member ASRB New Delhi, to address this August gathering. Sir, you are unmute, please. Chakrabarty, sir.
Yeah. Thank you, Dr. Amrita. Good morning, everybody. And the galaxy of scientists I could see. Dr. A.K. Singh, the most eminent director of IRI, one of the most eminent I have seen. And when I hear him, I see the subject which he speaks as if he is the master for all the subjects. And I'm very, very glad to see. We have seen Professor Anupam Verma and other who are there on the chairs. They are pathologists, definitely we expect, and as usual, they are the seasoned scientists as well. But Dr. A.K. Singh, I think he is the only, other than pathology director I have seen, heard, and on many occasions, be it drone-based management, be it clear field technology, be it this gene pyramiding-based thing, he gives great importance to this science or branch of botany, which we can say, which is very important. And thank you, Dr. Singh, for such a nice introduction to the subject. And in fact, my job part, a lot of it is done. I am also delighted to see Dr. Manohar Acharya, because when this program was conceived, the first person in the name of mycology today which came to my mind was Dr. Manohar Acharya. But Dr. Pramila also had been contacting him. Unfortunately, we heard that he was down with the COVID. So we were little, but then day before yesterday when I just called him, I saw him that he's there on this panel and he's there. Sir, welcome again and our regards to you. And there are a galaxy of other people. Dr. Anupam Verma, without mentioning, he's another stalwart I have seen. In fact, be it he is on several policy making bodies for biopesticides, for deciding or banning of pesticides, which pesticides rationally he has done very good, and that is a landmark achievement in the field of plant pathology. Of course, virology, without mentioning, cannot be addressed without him. Dr. Anupam Verma is there. I'm grateful to you, sir, and we seek your blessing and guidance all the time. Dr. Jalali, he's always known for biocontrol sciences, and he was the I know him for last several 3 decades at least, and when he was also director of NCIPM. Sir, your presence again encourages us a lot. Madam Usha Sharma, as usual, she's such a motherly kind of and sisterly figure, and she's a specialist in mycology. Dr. B.N. Chakravarti, as usual, I know, he's our guide here now. Many times whenever I have to immediately look for somebody for guidance. So I look upon him. He is there. Dr. Varun Bal, they're all very leading scientists. Dr. Saharan, Dr. Dubey, Dr. Yadav, Dr. Pun, Dr. Pant, Dr. Celia, I'm able to see, Dr. Vishnu, Dr. Miss Shesha. So many, there are, I don't want to just take time now. There are 47 participants have already there. I'm very glad that Delhi chapter of IPS zone has chosen to conduct symposium on a unique and much needed topic of modern trends and systematics and prospecting of fungi. I must congratulate the zonal president and Dr. Pramila and the zonal councillor Dr. Deeba for taking up this vital topic for delivery. This is very much important at this point of time. At many times, Dr. Manoharachari knows we wanted to keep this subject alive because world over, I was reading a paper like Dr. A.K. Singh said. I was reading a paper last night. It was showing that US and Western world is still bad, although they were the pioneers initially, but now they are wiped out of mycology. I was at one point of time when I was in the US, they were telling if you are a mycologist today, we are going to take you from your home to employ you there. But if you are a biotechnologist, we don't have a space or place for you. So I was very happy because this was a subject which was of prime importance when we started doing our MScs and PhDs. But now mycology is people again shudder to see this as a subject. Presently, 2.3 to 3.8 million fungal species, as already spoken by. This is the latest statistics are believed to exist in the earth, and of them, 1,48,000 of these species have been described and named. Importantly, just over 17%, 25,611 species are cultured and publicly available at different collections throughout the globe. According to Botanical Survey of India, 15,396 species of fungi exist in India. As per World Federation of Culture Collection, there are 32 microbial cultural collections in India which conserve more or less 36,000 fungal accessions in the country. These are the accessions which are conserved within the country, despite this rich fungal diversity in the country and a substantial effect or effort towards conservation. There were only 61 new fungal species were described in India in 2019, in comparison to 377 done by China and 129 by Thailand. So see, again, these countries, Asian countries, India is of course one of the most prominent country. We talk much about fungus because this is the second largest, I think, kingdom after insects. Well, so this is a very important, and there you all are dealing every day with the fungi and all. So this is one of the most versatile kingdom which we are talking about, and it needs to be preserved, protected, and studied very well. It is imperative that to approach a complete catalog of fungal diversity of the country, it will be necessary to dramatically accelerate the pace of species description, because at this speed of identifying 61 per year is very less, whereas only 7% or 6.7% of fungal species have been characterized so far. We have lots and lots of the things to do, because I have a reason behind telling them this point, because so far we have only 9 biopesticides registered. There may be millions and millions of them which can be exploited as a biopesticide, but if you see for Central Insecticide Board and Registration Committee, 9 microbial biopesticides are registered, out of which many are fungus, virus, some NPV and other things, and some bacteria, and including all 9 are there. So where are we? We have to go ahead and look for such for mining or whatever you are telling, prospecting. We have to see those kind of things. Fungi has been playing an indispensable role, not only as economically important plant pathogens, but are also most prominently used as a source of foods like truffles and mushrooms, and industrial products like organic acids, alcohols, antibiotics, penicillin, the very first antibiotic which was there. It came from the fungus. Growth promoting substances, which we now called as a one of them. They also have been included most recently as a biostimulant, because the guidelines have already been made by us, and it has been accepted by Government of India. It is going to be gazette notified very soon. Enzymes and amino acids and whatnot. Production of and fermented beverages, bread and vinegar are traditional processes. These are the fungi which are there, which are involved, practiced from time immemorial for early civilization. Many of these in probiotics as well as in case of fermented bakery products, they have been further modified. They have been engineered to remove the clinical elements from those, or clinical determinants of those fungi, and then they have been now tamed to make. So a world of or wealth of resources are available with us, which we need to exploit. And but mycologists, if we see in the country, you can count on the head, on your fingers, how many mycologists are there. But then in agriculture, fungi are playing significant role. Again, these are the industrial uses which I used, and food use. In agriculture, conjointly they play physiological process as well as mineral and water uptake, like VAM. Then many fungi live inside the plant, and this is another whole lot of a new subject which is being looked upon. We have a 4 crore worth of project with NBAIM on endophytes. Endophytes, many fungi, they live inside the system of the roots of the plant system inside. They are the same fungi as fusarium and some other species which cause disease when they are outside. So we need to study the system of taming them. The same fungi which otherwise many times cause disease, when they are inside and they live amicably inside the endophyte, favoring the host. Like many of the causes of resistance to pesticides, heavy metals have been ascribed due to these endophytes which are present, which gain entry into the root and they work like commensals. So what are the mechanism of those things? Now we have come to an era where fungal morphological based this identification will not be sufficient. Prospecting needs to be done for everything. For human genome, we have sequenced. We are sequencing many of the pathogen sequences. Fungi are the major decomposers of the dead plant matter and are the major constituents even for the stubble decomposers, which recently either Delhi government or NBIM has been using. So they are the components of that. and of plant production and agricultural waste. Their role as biostimulants in growth promotion and on the biopesticides for management of pests have resulted in exploiting their use as industry that is growing at an annual growth rate of 12 to 14%, much faster than the chemical pesticide industries, which is growing merely at the rate of 8%. So this is the time. This is the time when world is so conscious about the use of chemical pesticides. We have to make judicious use of. Even I was surprised to see many of you know Professor Charudatan. He has used. He has opened up a company for biopesticides, and he has commercialized and registered first virus for control of tropical soda apple, that kind of weeds in the United States. And so why not we have very eminent virologists here talk about. I mean, this again other than this, I am just telling about biopesticide. So biopesticide, we have fungus as a bionematicide, herbicide, and then mycoherbicide. So recently, one of the company within India, and the person is very well known to me, and he has obtained 21 smart formulations of mycoherbicide. He has obtained 21 patents, not only in India, but we are yet lagging behind. We are still working with the active fungi, which we do not allow many times to import because of bioterrorism and other things, related reasons. So the time has come when we have to focus on the bioprospecting and the genes which are responsible for as a biocontrol determinants. So these things needs to be done. Their expression has to be done, and cell-free cultural filtrates expressing these gene products. These are the one which are smart formulations, which are there. We no more appreciate the use of bioformulations or biopesticide formulations which are there with the active fungal propagules there. So why not identify the 2 or 3 gene products in every biocontrol agent which are expressed in a cell-free culture filtrate, and such kind of formulations neither can be there can be IPR pilferage, and we have to start initiate working on that development of bioformulations which are smart formulations. The industries have started doing. So why not this intelligentsia which is sitting here and which is contributing to this size. Despite this, only 9 major microbes have so far been, as I have already said. Thus, mycologists must play a major role in identifying and characterization of such microbes that can be exploited for biointensive IPM protocol for organic culture. The pace of identification must be enhanced manifold. Phenotypic, genotypic, prospecting, smart formulations are top of the day. And tomorrow, nobody will say that I will be spraying or applying in the field the live cell of trichoderma. They will be seeing, because a trichodermal species might be having 30,000 genes, but hardly 4 or 5 genes are important for which biocontrol activity is seen. And that will also remove the concerns of Central Insecticide Board and Registration Committee, and that it has to be tested for some toxic clinical toxicants and all those things. Because other genes are there which are allergenic and all, for which Indian Institute of Medical Sciences or All India Institute of Medical Sciences or ICMR, they insist us that none of the strains of trichoderma can be ignored without or can be given registration, even if 99% are non-toxic. Some of them may have some allergens. So every new strain is subjected to this thing. So if you have a smart formulation, you have a biopesticide or bioformulations with having expressed genes for 2 to 3 genes which are antagonistic, this thing imparts that antagonistic property. So nobody will question that, because those are the ones which might be chitinase or cellulase or something which are not a issue for clinical concern. Now, this is about the role of fungi and which needs to be done, which needs to be exploited. Now, coming a little bit about the taxonomy or systematics part of fungi, world over mycologists and taxonomists or fungal systematicians have become an endangered commodity. I'm sorry to say, but this is such an important path we have come to stage. I learned mycology from a famous mycologist, Dr. R.L. Munjal. Sir, many of you know that Dr. R.L. Munjal was there in Solan. I studied my mycology. And I take pride in telling. And subsequently, he came to IRI, and I studied mycology from him, and I was very favorite student of him. In fact, I thought I will be just going into mycology, and even I know, I remember the entire book of Elaxopolus and Bisbee at that time, when there were five major whatever orders or something, Mastigomycotina, Eumycotina, and all those things were there. So I still have that confidence today that I can still see from the fruiting bodies or some kind of other signs and symptoms. I can see what are the fungi to which it belongs. So he was such a taskmaster. I will just say a small thing. Uh, when he used to give practicals, only when we used to do in Solan, practical 10 out of 10 I got, because when I went inside after the theoretical part, after when I went inside to his room for practicals, he kept some fungal species and all those things, some in microscope I could identify, and then he suddenly he caught my hand, and he said, I had given you that sample from forest. Have you identified that? I said, yes, sir. And I said, I cultured and I isolated. So suddenly he was on the other side of the table, and my hand was there. He caught my hand, and he just inverted my hand and saw my hand, and he was happy, and he gave me 10 out of 10. I was wondering what he was seeing. He was seeing that these hairs should be gone, according to him, because when we used to hold over the flame inside the tube when we are putting, so these hair used to, this is his way of thinking. And then he thought, yes, he has really done the practical. So that was the type of task which they used to do earlier. But I am little bit afraid now whether those kind of things are being followed with the advancement of science. um, Mycologists, they must play a major role in identification, characterization of more such microbes that can be exploited for biointensive IPM. That I said I learned, uh, and India has produced a world-class mycologist like Dr. J.F. Dastoor, Dr. L.S. Subramanian, Dr. B.K. Bakshi, Dr. Thind, Dr. C. Manoharachari, who is sitting here. And these are the people whom we hail, and many others, definitely. Now, Dr. Pramila, Dr. Usha, all of you who are sitting here. So you are a very essential commodity, and we need to see that how your efforts and their efforts have paved way for the modern mycology, who have spent most of their lives for developing this interesting discipline in India. Today, a world need many such kind of devoted mycologists in department, perhaps Dr. Pramila and Dr. Diva, Dr. Rashmi, and very few maybe people are actually involved in mycology, although most of us are dealing with the fungal diseases, but not into the systematics part of the things. In their absence, and due to lack of interest and patience, many universities where plant pathology used to be an integral uh, Department of Mycology and Plant Pathology, word mycology has been dropped. So this is another pathetic situation in the sense, this is sad. This is sad. In fact, I felt when Solan, it was mycology, plant pathology. Even in IRI, it was mycology, plant pathology. Yesterday, I was looking for an expert in Birsa Agriculture University. They still have mycology and plant pathology. I was very happy because those are the places where such, and many of you are from there. They are very familiar with that university. From although there is a scientific reason behind dropping the word that plant pathology involves pathogens of various nature, nevertheless, After insects, fungi, as I said, perhaps constitute the second largest kingdom and need special attention. So mycology has remained merely as a subject, but when mycology and plant pathology used to be there, mycology used to get a bigger status. And but that does not mean that virology is not important or some other bacteriology is not important. Same is the fate with the systematics for bacteriology also. Future of fungal taxonomy, as Dr. A.K. Singh and systematics is very challenging now. Traditional morphological studies are still very important, as species name, as he said, are very important. Therefore, taxonomists must, but since the advent of molecular sciences have come, taxonomy must incorporate modern techniques as phylogenetics, genomics, metabolomics, evolutionary approaches. This is to make it more meaningful than what we didn't used to have earlier. Otherwise, mycology was being done otherwise also. Now, we need to exploit these studies. IRI is maintaining an imperial culture collection of nearly 4,000 fungi, and many of them have been revived at NBAIM also, and they are still being maintained at both the places, I believe. And but these are the excellent sources of studying the evolutionary studies of present day strains of same species, which might have developed. Because you think of the time when DeBerry and Butler, when they have collected 50,000 samples of disease herbarium, which is still there in Imperial Agriculture or Indian Agriculture Research Institute. So those are the excellent sources which might have developed as a result of selection. Now the strains of the same species might have developed, and they have undergone pressures, selection pressures of external stimuli such as climate change and other exigencies, giving now a clues to the mechanism of their evolution and possible management. So those are the very important resources. We need to protect them, preserve them, and see. But at the same time, we need to see preserve them in a scientific manner, that they should not lose their. They might have lost, some of them might have lost their pathogenicity or original virulence pattern, but still they might have certain genes which might lead to certain evolutionary trend of the new strains of the same fungi which existed now 70 years hence. On the other hand, although the molecular tools have aided in the identification of fungi, but it has definitely deterred the interest of scholars towards traditional taxonomies. It must be understood that molecular tools alone cannot be sufficient to describe a new species, as correlation, hyphal structures, mode of reproduction, etc. will always be required. It is pertinent to mention that despite engagement of significant research personnel, scholars and students throughout the country, only a few institutions and organizations like BSI, Botanical Survey of India, NFCCI, which is Agarkar Research Institute, and a few universities have successfully described, maybe IRI also, have described new fungal genus and species. So this needs to be strengthened, and everywhere we need to expand this area. This clearly indicates the lack of trained human resources for traditional fungal taxonomy, although use of molecular tools has become routine in almost all research organizations. For giving better meaning to traditional taxonomy, computational mechanisms are required to be identified for better resolution. So this is uh, chipping with the modern technology now. Now with the taxonomy, morphological and other things, now how we take the advantage of latest technology which are coming. Metagenomic sampling and sequencing needs to be done. We have now many of the fungi which are not identified because they cannot be cultured, but then metagenomic studies and clues for their genomic and other constitution can be done. Integration of genomic data with fossils, ecology, physiology data is required, and systematic integration and metagenome data with genome derived phylogeny needs to be done. A significant reorientation in academic curricula and research priority is urgently required to revive this discipline. So these are the points which I wanted to talk, and this is a subject very close to my heart, although now we are dealing with the applied aspect of the thing. But few persons like Dr. Manoharachari and some of you, Dr. Pramila, and those who are presently working on systematics, you are very important. But you have to give your best for this, to do something for identification in your lifetime. I think this is the one, when you are here, you have to see that how you identify more, at least 100 fungi you identify, a new one before you lay down the office. You have long time to go. A significant work has to be done. I wish this symposium a great success. And when I see the entire program, I feel very happy to see that it revives all the things which needs to be studied in mycology, and it has all the elements of that. And so I wish you all a very success. And at the end of the day, if you develop some niche area excellence, Professor Anupam Verma, Professor Jalali, Dr. Manoharachari, and many of you, sir, we learned things from you, all the subject from you. So if we can develop some niche area of excellence for each of these propagules or pathogens, for virus, for fungus, for phytoplasma, which are coming up new one, and many other bacteria, which is again another very important species, economically important. If we can have some niche area excellence and give some money and create some bodies for strengthening the studies of mycology in India, because particularly world over now, people are saying that mycology is developing only, it is contained now only in Asian countries. So, and in Asia, we are the largest, what to talk about Asia, in the world, we are the largest, and we have a largest diversity also. So we have several other things, unfinished work. So we need to give a fillip to such kind of studies only by creation of a special boost to this subject. And I wish at the end we have a series, some of the recommendations. Already as a president of IPS, I said with all these zonal conferences and special conferences, special meetings, we need, all of them need to generate something unique recommendations which will find place in the booklet, which will be placed in front of a policy maker, but where we need to give research policy, where we need to create some other related policy for the development of agriculture in general. Thank you so much. Thank you, organizers, for giving me time. Thank you so much.
Thank you, sir, for your informative speech on the scope of mycology field. Now I request Dr. C. Manoharacharya, Scientist Emeritus, Osmania University, Hyderabad, for his valuable speech.
Sir, kindly unmute yourself. Amrita, you can unmute him. Host can also unmute him. Yes. It was just done and again it got muted. Yes.
Okay.
Yes, sir.
Okay.
Dr. P.K. Chakravarti, a visionary and a well-known policy maker and plant pathologist of the modern era. Dr. Rashmi Agarwal with 3 feathers on her cap and rust specialist and with a human touch to the faculty and all the plant pathologists. And we have Dr. Pramila Devi, Zonal President, and Dr. Gagoi, Secretary of IPS. And we have dignitaries like Professor Anupam Verma, Professor Jalali, the great teachers and eminent plant pathologists, policy makers, and who have nurtured the Division of Plant Pathology and universities. And I have got a lot of respect for them. And Professor S.C. Dubey and Professor Chakrabarti and all other IPS executive members and Usha Sharma madam and every one of you, my Greetings and pranams to every one of you. I thank Dr. Pramila Devi and Dr. Gagoi for having given me this tiny fungus an opportunity to be with you. The topic that has been selected is appropriate, and the modern times needs such kind of topics. That is number one point I would like to make. The second thing is that biodiversity and systematics and their prospecting is an important aspect, and this particular topic has been chosen by them, which is again a significant point to be noted. The third one is Hawksworth and Licking made an estimate in 2017 that there is 1.5 to 3.8 million of fungi in the world. That is the estimate only. And then later on, Wu et al. In the year 2019, they have come out that there is 14 million of fungi. Now, to make an attempt to identify even 3.8 million of fungi up to the species level, it takes more than 4,000 years. And if it is 14 million of fungi, I doubt that whether the mycologists will be there to identify those fungi by after 10,000 years, because everybody is after biotechnology and molecular biology. I'm very happy that Dr. Chakravarti and Dr. A.K. Singh made a mention that mycology needs a lot of boost and it has to be sustained. But my point of view is that as long as agriculture sustains in our country and elsewhere, mycology and plant pathology will definitely sustain. Without mycology and plant pathology, no agriculture can sustain. It is a fact, whether they agree or not, but one has to give importance for mycology and plant pathology. And Professor Jalali sir has been an ardent supporter of mycology, and P.K. Chakravarti and Professor Anupam Verma and others, and they have been helping the institute for the growth of the mycology, and they have been supporting. Now, if the 13 million or 14 million of fungi needs to be identified, what will be the scenario in India? As on today, 120,000 species of fungi have been identified in the world. Now in India, we have different estimates, but my estimate exactly is 29,000 species. And because many of these agencies which are there, Ministry of Environment and BSI, they did not take into account the remote papers that have been published elsewhere, and that they have not taken into consideration. Therefore, what I would like to say that 29,000 fungi is the estimate. Identified. Now, as on today, the estimate is around 2 million fungi for a tropical India. And if it is 2 million of fungi to be identified in India, then naturally, how many mycologists we need? Kindly please estimate. So mycology has to be given a boost, and mycologists have to be appointed in different R&D organizations, including the case of IRI, Division of Plant Pathology, and many other institutes. So there is a necessity for such kind of appointments and boosting of the mycologists and mycological research. Because from the point of biotechnology, fungi contribute to $16 billion worth of biotechnological products. You take any product from all over the world, it is the biotechnological products. You take mushrooms, it is a multi-billion-dollar industry. Nobody can deny the fact. And even in IRI, since the time of 1907, Butler, and then till today, many eminent mycologists served this particular one. And they have seen the growth of different branches, including plant virology and as well as the plant pathology. And that is the essence of the mycology. And they supported plant pathology, they supported plant virology, bacteriology, and new branch of whatever it is, mycoplasma and phytoplasma, so on and so forth. So all of us should support the greater aspects which are going to help the humanity. That is the role of a mycologist and plant pathologist. Now coming to different levels of understanding of mycology, I can say that high throughput sequences of has helped us to understand culture-dependent microorganisms, including fungi, and as well as the culture-independent, that is non-culturable fungi also. And this has given a clue for us to go for the metagenomics and other things, and that has yielded a lot of fungi, including the identification of the cryptic species. I'm very happy to say that the culture center at IRI is doing a very good job, and herbarium in no other place in the case in India they are maintaining any herbarium. It is only IRI division of plant pathology which has been maintaining the herbarium, and that is an important aspect which needs to be taken into consideration by the policymakers, and therefore it is a must for us to have only 7% of fungi have been cultured. If you want to have fungal biotechnology to grow and to have a market, greater market for the secondary metabolites to become medicines and so on and so forth, and growth promoters and other things, biopesticides, there is a necessity that one has to culture these fungi. And if the cultures are made available, that will be a boost for any kind of industry. So we must make an effort to make, at least we should make in India that when there is 1/3 of global diversity of the fungi, that is the hidden wealth that is there in India, we have to go for in search of the fungi, including the extremophilic fungi, which are very, very important from the point of industry and agriculture and medicine. Therefore, this is an aspect to be taken into consideration, and that is how I consider that mycology, systematics, and advances biotechnological aspects and other things have become very important from the point of fungi. Now, since 1907 till 2010, the mycologists have played very important role, and they discovered, based upon morphotaxonomy, they have discovered a number of species and genera. India has contributed a lot in that respect. But unfortunately, after 2010, CBD and National Biodiversity Board and Melbourne Code and the nomenclatural rules have made very difficult for Indian scientists. As per the Melbourne Code, they say that they have to deposit cultures or material in 2 cultural centers, that is culture deposition centers. And CBD and National Biodiversity Board doesn't allow Indian mycologists or Indian scientists or Indian plant scientists to deposit them outside. Nor they will allow the others to deposit them in India. That is the drawback. And after 2010, if you take a data, then not many genera, new species and new genera have been described as on today. Therefore, scientists have gone to patent their fungi outside India. They have gone to Germany, they have gone to America, and they have given their cultures and other things to some other way, what I don't know, but they have become very important from the point of view. And many of the biotechnological products, fungal biotechnology products have come out of the patents and other things they have commercialized. Unfortunately, I have to tell the policymakers like Dr. P.K. Chakravarti sir and Dr. Anupam Verma, Dr. Jalali sir and others, sir, in DBT and DST and other organizations, many of the patents are lying there. Please advise them to make them commercialize. Otherwise, all our Indian cultures, all our Indian patents will go waste until and unless they take care of them and bring them to the form of a product, and see that agriculture scientists and as well as industrially, from the point of industry and as well as from the point of medicine, and all these things will be solved in our country, including the recent aspects of the disaster management, health management will be taken care of by the fungi and other microbes. This is an important aspect to be dealt by the policymakers. Now, coming to the various other things, for example, you take Ophiocordyceps sinensis. You see that $58, around 20 capsules of the Ophiocordyceps. It is called Indian Viagra, and it is anti-cancerous drug is extracted from that. There are 7 species in India, and none of them have been cultured. Termitomyces, an edible fungus, which has got all 18 amino acids and lot of protein, and that has not been cultured so far. And we have so many such kind of fungi which have not been cultured. Even rust fungi, Madam Rashmi Agarwal is here, rust fungi have not been cultured. They have to be maintained on the plant itself. So that is, these are some of the issues about the non-cultural fungi and culturable fungi to be taken care of. Ophiocordyceps is now costing for $58 20 capsules, 1,000 rupees for about 10 grams of Ophiocordyceps or 20 grams of Ophiocordyceps. That is the beauty of the fungi. And we have 30,000 of that particular mushrooms around the world, and multi-billion-dollar industries elsewhere. China, Brazil, and then Thailand, they are making the roadways and highways for the mushroom growth and as well as for the fungal biotechnology and fungal taxonomy. And why we are not able to make it is, and rightly it has been pointed out by Dr. P.K. Chakravarti and as well as Dr. Jalali earlier, that mycology needs and mycologists need a lot of support. And it is very clear that taxonomies have become endangered species. But at the same time, I must say that it has become tax on the youngsters. Youngsters they are not showing interest towards the taxonomy, and they are not sitting with the microscope. They are not going. First thing is morphology is very important. Say, you see, taxonomy is a way of life. Taxonomy is the mother of all sciences. If you forget that no science, if you forget taxonomy, no science will exist in this world, particular world. So taxonomy has to be taken care of by everyone, and one has to search in. They have to go in search of many new fungi which are going to be for the human welfare in near future. So taxonomy and maybe become endangered species, but now there are only around 20 taxonomies in India, and lot of that is workshops and other things have to be organized to train the youngsters, and they have to be, interest has to be inculcated among all the interests by saying the importance of this particular group of fungi and as well as how they are going to help. And now the anti-cancerous drug, and maybe in near future it may solve many of the problems also. So in other words, I must say that, and many good points have come out of all other speakers. I congratulate all of them. I also thank the organizers, and I must place on record for the presence of the various researchers and the faculty of Division of Plant Pathology, IRI, and all the members, and also the members of IPS Delhi Zone and a few others and the dignitaries. And at the same time, I also say that a lot of work has been done at IRI.
I think the net problem is there.
Yeah, yeah.
For a meeting, and that is how the ICR and they are doing very well. I thank every one of them. And last, I would like to say, train PhD students to be thinkers also, not only only specialists in their field. No fungi, no life. Thank you very much. Thank you very much.
Thank you. Thank you, sir, for your valuable notes. Now, I would like to request Dr. Anupam Verma, Adjunct Professor, Division of Plant Pathology, IRI, New Delhi, for his valuable speech.
That was not in the program, my dear.
So you can give some remarks on this. Please.
Well, I would only say that I'm delighted to hear from Dr. Ashok Singh, Dr. Chakravarti, and Dr. Manoharachari. I'm sure if Jalali Sahab was allowed to speak, or maybe speaking later, will say the same thing. Mycology requires strengthening. Now, just because you have given me this platform, give me a couple of minutes.
Please,
sir. At one time, all botany departments of the country had mycologists. And gradually, that changed. The practice did not continue. Manoharachari is an exception. That he's continuing, and he continued right from the beginning. I've been associated with him I know for now more than 40 years. At one time, you people may not be knowing, at ICR, there used to be uh, a group of people, some name was given there, who were doing planning and approving projects. There was a group of plant pathology which included mycology. And at the time when Dr. Mehrotra of IRI was the ADG, and I was a member there, I gave in writing that what ICR should do is to have a specialist in mycology for particular group. For example, mycology is such a vast area. Promila cannot do every group. Diba cannot do every group. So we must identify across the country the people who would be handling a particular group. Dr. Manohar said IRI has done very good work on culture collection, and also our herbarium is actually the only herbarium of this nature which is being maintained. But I can tell you two things here. Number one, that we try to strengthen this because we saw, Dr. Munjal was there, Dr. Kapoor was there, Dr. Sarbhoy, and we had a very good backup team, Ms. Lal, Dr. Lal. They were working. We wanted to strengthen that. In fact, we wanted to make this as an advanced center for mycology because every area, similarly, an advanced center for bacteriology, but here we are talking mycology. Dr. Manohar, just for your information, I would like to tell that I was instrumental in making mycology as a compulsory option for the students who join MSc. I think Rashmi will confirm.
Yes, sir. Yes, sir.
Okay. Now every batch, if it is a batch of, because normally we were taking 4 or 5, at least one had to. Unfortunate thing was that most of the years that one was the last in merit because it was forced on him. I'm happy that some of them continued with mycology. So that's a hope. In fact, this seminar, Pomila, thank you very much. Rashmi, thank you very much for giving me. I'll discuss with you. I have to write something about mycology.
Definitely, sir.
Because mycology needs to be strengthened. Everybody, Dr. Monachari has made that information about patenting. And we don't have time to discuss. It's very important.
Very important, sir. Yeah.
And I can only tell that I wanted microbes to be patented in the country, and I made that presentation in the parliament for making microbes patenting. One very senior scientist there asked me if the patenting is done, then how much money India would have lost because they had to pay for patents for others. Because all those drugs which are coming, many of them are actually of Indian origin. Okay? I think with this, I would say mycology needs to be strengthened.
Yes, sir.
And I'm sure with the group, active group here, it will be done. Thank you very much.
Thank you, sir.
Thank you. Thank you, sir.
Thank you, sir, for your valuable words. Now, I would like to request Dr. Jalali, sir, ex-director of research, Haryana Agriculture University, for his valuable remarks. Sir, please unmute yourself.
Unmute करिए sir, unmute करिए.
Sir, please unmute yourself.
Sir, Jalali sir, unmute please.
Unmute.
Well, thank you very much. I think much has been said, and there is hardly anything to be said now. In fact, for the last several years, we have been campaigning at all platforms about the bankruptcy with the discipline of mycology and taxonomy in general, and mycology in particular has gained. And this has really caused a great vacuum in the correct diagnosis and subsequently the management of dwarf identity. I'm just surprised. Kindly allow me to give my voice to my inner vibration. All said and done, one of the most premier institutes in the country is IARI. Excuse me. It used to be a division of mycology and plant pathology. How come the word mycology has been dropped? Now, I don't want to raise an issue now. I have raised earlier as well at many platforms. Nevertheless, this is high time that we still should gain qualitative momentum. Otherwise, in fact, the time is already there. It has already arrived that people are not able to diagnose or even identify fusarium from helminthus folium. Many PhD theses have been submitted with wrong nomenclatures on the title. I'm sorry to tell you this. Couple of PhD dissertations have been rejected because of these misnomers made on the title page. Now, we have been saying so many things for the last more than 1 or 2 decades. I'm sure most of these legendary figures which I see on the screen here have been witnessed. I remember the great mycologist A.K. Sarba, for example. We used to raise lot of issues. There was an issue about raising a Commonwealth Institute of Systemic, if I recall correctly, couple of years back. Nothing very tangible has come. The ball stops always at the policy level, and nothing, is really happening, with due apology. Nevertheless, in recent years, I see some ray of hope. And one of the examples is definitely IARI, where they have been baptizing the molecular tools now in addition to the morphological tools. And I'm glad that Pramila Devi and her associates have also embarked upon harnessing certain, like DNA barcoding, for example. I have been visiting their center virtually every time when I visit Division of Mycology Plant Pathology. Rather, Division of Plant Pathology now, of course, IRI. And I have seen, as has been said, that herbarium is the only one here in the country. And every time when I go, I see a lot of change, qualitative change. And I think I must congratulate them for having given this initiative. DNA barcoding of many of the fungi have been done. Diba, for that matter, along with Pramila, they have been doing good deal of work in this domain. I could see they have done on Trichoderma, if I recall correctly, Pitomium, if I recall correctly, Curvularia, and so on and so forth. Now, all these exercises have been done. Nevertheless, as has been said by Professor Manohar Achari, lot needs to be done. Otherwise, the vacuum is so big that all these little efforts here and there does not match. And therefore, it is again, in fact, I'm tired of telling all these years. I have used a lot of, exercises in convincing the younger, and partly I would hold Professor Manohar Achari responsible for. He has not been able to raise issue with a greater voice except giving these lectures. In fact, he is one of the very few now left in the country whose word in the world of mycology in India can carry a huge weight. And I must really appreciate the effort done by IPS North Zone Delhi Zonal. And Pramila and Diva, they have highlighted now the thematic area of taxonomy and inviting Manohar Acharya. And I am very keenly looking forward to listen to his keynote lecture this afternoon. Well, thank you very much and wishing you all the best of luck and success in your future mycological endeavors. God bless you. Thank you.
Thank you, sir, for your valuable words. Now, I would like to request Dr. Diba Kamil, Zonal Councillor, IPS Delhi Zone, and Co-Organizing Secretary to present vote of thanks.
Thank you, Amrita. A warm and cherished morning to all most valuable, our most valued honorable chief guest of honors, distinguished guest, very senior IPS members, IPS Secretariat, Organizing and Technical Committee, qualified scientific staff, and everyone. It's my privilege to propose a vote of thanks, acknowledge the contribution of those who worked hard to make this IPS Delhi Zone Symposium inauguration function successful. I am very grateful to our director, Dr. A.K. Singh, for his kind acceptance to be the chief guest of this function and his valuable suggestion all the way to conduct this IPS Delhi Zone virtual symposium successful. I am really thankful to the guest of honor, Dr. P.K. Chakrabarty, sir, President IPS and member ASRP, for his continuous support to IPS and especially for this Delhi Zone symposium. I am really very thankful to our guest of honor, Dr. C. Manoracharya, sir, to attend this symposium and give his expert observation on the fungus systematics and especially taxonomy. I want to thanks to our Dean, Dr. Jashmi Agarwal, ma'am, and she is also our beloved head division of plant pathology for her motivation, support, and taking so much effort to make this program successful. I'm very thankful to Dr. Robin Gogoi, Secretary IPS, for his all-time support and his friendly approach towards us. I'm really very thankful for the bottom of my heart from my all seniors, especially Dr. Anupam Verma sir, Dr. B.L. Jalali sir, Dr. Usha Sharma ma'am, Dr. B.K. Chakrabarty sir, Dr. S.C. Dubey sir, Dr. Shilpi Sharma from IIT Delhi, and all, which I'm not taking all the names, for they happily agreed for joining this meeting and grace this occasion. I special mention is our vulnerable IPS Secretariat, Dr. K.K. Mandal, Dr. Malkhan, and Dr. Raju for their support. I lack word to give my gratitude to my mycology team, Dr. Pramila Devi, ma'am, Dr. T.K. Baksar, Dr. Amrita Das, and Dr. Divakar Bahukandi sir to attend and give all the support to conduct this meeting. I want to thank all the members of the technical committee of this symposium, organizing committee of this symposium, and all the scientific staff from IRI and especially Division of Plant Pathology. Dear esteemed guests, ladies and gentlemen, once again I would like to say that we all are most grateful for the entire participant of this forum. We thank you to being with us this morning. It's great pleasure. Now, thank you so much. Now I am inviting Dr. Robin Gogoi sir from the IPS for further. We have a condolence meeting today of Dr. Ashok Gaur. Robin sir.
Is the slide visible?
Yeah. Yes, sir.
Slide is
yes,
visible.
yes, it's visible.
So, respected all the members of Indian Phytopathological Society, one of our beloved senior member of this society, Dr. Ashok Gaur, passed away 2 days back. So, to commemorate his demise, so we are going to hold now one condolence meeting. So, we are really very saddened after hearing the news that he is no more. Just he passed away on 14th December. And just I will read out that condolence note will be sending to his family. The society is distressed to hear about sudden demise of Dr. Ashok Gaur, ex-principal scientist in the Division of Seed Science and Technology, IRI, on 14 December 2020. Dr. Ashok Gaur was born on 3rd July in the year 1945 in Allahabad in a family of teachers. Dr. Gaur did his MSc in 1967 from Kashi Naresh Government PG College, Gyanpur, Varanasi, Agra University. After MSc, he worked as research assistant in CSIR-sponsored scheme on mosses of Kumaon for a short period. In the year 1969, he was selected for the post of research assistant in the Division of Mycology and Plant Pathology, IRI, New Delhi. In 1972, he was selected for the post of research assistant in the Division of Seed Technology, which is known nowadays as Division of Seed Science and Technology. From 1972 till his retirement in the year 2007, he worked on various aspects of seed pathology. His main area of research was storage of treated seeds, revision of certification standard for loose smut of wheat, and chemical control of seed-borne diseases. He also provided Hindi equivalents of the name of various plant diseases, which were compiled as Compendium of Plant Diseases, published by the United States Department of Agriculture. He was fellow of Indian Society of Seed Technology and Indian Phytopathological Society, and member of many professional scientific societies. He was the editor and chief editor of the journal Seed Research, and editor of Seed Tech News, published by Indian Society of Seed Technology. He was the zonal president of Delhi Zone Indian Phytopathological Society in the year 2003. So we from Indian Phytopathological Society would like to extend our heartfelt condolences and pray to the Almighty God for eternal peace of departed soul, and also pray to provide sufficient strength to the bereaved family members to bear this loss. Om Santi, Om Santi, Om Santi. Thank you. Yeah. Over to Amrita.
Okay, thank you, sir. Now we are entering the first technical session of today's program, which has the presidential address and M.K. Patil Young Scientist Award lecture. And the technical session will be chaired by Dr. Rashmi Agarwal as chairperson and Dr. Dinesh Singh as co-chairperson. The rapporteurs are Dr. Malkhan Singh, Gujar, and myself. So, ma'am, Rashmi, ma'am, now I request you to kindly take over the session.
Thank you, Dr. Amrita, and I welcome all of you on this first technical session. As Dr. Amrita mentioned, that this constitutes presidential address and one award lecture. Before I begin the technical session, I once again welcome all of you, and I congratulate Dr. Pramila and her team for very nicely organizing this Delhi symposium, very meticulously planned. So we'll begin this session. Dr. Dinesh Singh is here. Has he joined, Dr. Dinesh? Yeah, he's there.
Yes, ma'am, he is there.
He's there.
So I would just like to mention here that I also have to join the Extension Council meeting. I'll begin this session, and in between for a while I will go there for half an hour. Maybe there are some from the division side. As head division of plant pathology in the Extension Council, my presence is required. So I will begin this session, and I warmly welcome all of you and kindly be there during this whole session. I welcome Dr. Dinesh Singh, co-chairman of this session, and also rapporteurs, Dr. Malkhan Singh and Dr. Amrita Das. So first is a presidential address. For this, I invite Dr. Pramila Devi. As mentioned, Dr. Pramila had been taking mycology, shouldering all the responsibility of ITCC and herbarium together. She's very meticulously and very methodically working in the area of mycology, taking all the team members together, maintaining, supplying, identifying the culture, and recently they have taken up bioprospecting also with the very efficient team members, Dr. Diva Kamil, Dr. Bag, and Dr. Amrita Das, and also with technical support. Whatever support can be given from the division side, I always give them, and all the other scientists always envy. They always say, ma'am, you are always looking for mycology strengthening, not looking into the others. But it is my small, humble effort only, looking into the meager resources we are having during this period. So with these words, I invite Dr. Pramila Devi. She will be speaking on conservation systematics, prospecting, and sustainable use of fungal biodiversity. Yes, Dr. Pramila.
Thank you, ma'am. Thank you so much for your nice words. Are you able to see my slides?
Not yet. Slide share. You had already opened and minimized your PowerPoint?
Open. Just open. Slide share. I have
you click on slide share.
Share screen, ma'am.
Share screen है ना नीचे share screen green.
Share the screen.
I did
green.
Yeah, I did it.
Then you double click on your PowerPoint, which is coming on the screen.
Yes, it's coming now.
Yes. It's coming. Go to the slide mode, the presentation mode.
Yeah. So.
Yes.
Thank you, ma'am. Thank you so much for your nice words. And for today's lecture of mine is conservation, systematics, prospecting, and sustainable use of fungal biodiversity. And yeah, as we all know, 1/3 of our fungal biodiversity lies in India. But for biosystematic studies needs for the essentially accurate and authentic identification, which includes the description of genera and species, and then formulation of diagnostic keys, establishment of phylogenetic relationships, and taxonomic revisions, and so on. So for this, morphological identification is very much essential. Earlier we were doing only the morphological identification, but now we are advancing with the advancement of this science. We are getting this molecular biology also, and it will resolve some of these biosystematic problems. But by only molecular identification, we cannot resolve the problems, because sometimes the morphology doesn't match with the molecular identification. So one of my students, he said, madam, morphological identification is very tough. Then I said, why do you say like that? And so morphology, molecular identification will be done by the machines, whereas the morphology, the human brains have to do. So then I realized why these youngsters are feeling very difficult for this, because it needs time and also the patience to do the morphological identification. But anyway, the authentic identification needs both morphology and molecular. So this for today's world, but the focus on biodiversity and bioprospecting, this is another area of using this cultures from the different culture collections. So on the preservation depleting biodiversity, so the culture collections play an important role. So for that purpose, we have the culture collections, which are the largest and oldest culture collections of India, lies in the division of plant pathology, IRI, Delhi. This is a Herbarium Cryptogame India Orientalis, which was established by E.J. Butler in 1905, and we are maintaining more than 50,000 fungal diseases specimens in HCIO, and the 570 new species and 19 genera were described from HCIO from HCIO. And then another one is where live cultures are being maintained is Indian type culture collection. This was established in 1936, and we are maintaining here more than 4,000 fungal cultures and 600 bacterial cultures. Recently, we have added in Indian type culture collection. ITCC is a member of World Federation of Culture Collections, and it is registered in World Data Center for Microorganisms. And recently, the Central Insecticide Board, which comes under Directorate of Plant Protection, Quarantine and Storage, has made it mandatory to deposit the bioagents before they are released for the commercialization. So the accession number is also in some of the journals is necessary from the ITCC for the publication. And so the ITCC and SAO scientists are actively involved in these research programs, and using both classical and also the molecular approaches. As I said, only molecular approach will not serve the purpose. So these are the, our centers like HCO and ITCC here to act as a national facility for maintenance, supply, identification services of indigenous and exotic fungal and bacterial cultures of various habitats. And we have a common activities of these HCO and ITCC, that is conservation, preservation, maintenance of cultures and also the specimens, and identification, supply services, and also the deposition services, and taxonomic investigations and documentation of fungal specimens as well as fungal cultures. Here you can see the preservation, how we preserve these fungal specimens in herbarium, HCAO. Here we keep the specimens in the folders with all the information, and we maintain them in the compactors. Here we preserve them this way, and we are preserving these herbarium specimens since 1883. Here you can see one specimen of even 1883, and this is the specimen which was deposited by E.J. Butler. So this way we are maintaining all these specimens since long time. And coming to the live cultures, in ITCC, we are maintaining them by different cultures, because we should not lose the culture. That's why we are in different preservative methods, we are maintaining them by repeated subculturing, paraffin oil, and also lyophilization. Recently, we have also started preserving in the 5% glycerol. And coming to the services, this is for the information of these youngsters, students. And here we do the service of identification of both fungal cultures and bacterial cultures. And we also supply the pure and authentic cultures for the research programs. And also we also get the deposition of your cultures and give the accession number after authentic identification. And all this information is available in our catalog, which is online, and this is the URL of that catalog. And as I was telling you, the molecular research we have started, but till 2007, we were only doing the traditional taxonomy. Then with the fund we got in 2008, we could establish the molecular mycology laboratory in the Division of Plant Pathology, IRA. Since then, we started working on both morphology and molecular together, so that we can get authenticity of the taxonomy and the identification of the any fungus. So we started with this trichoderma, genus trichoderma, and we have taken some isolates and then did the morphological identification, and we grouped them, and also we did the molecular identification of the same isolates, and we compared with the morphological. Wherever the morphology and molecular are matching, that we have taken as an authentic identification, and we have done the molecular, established the molecular characters of these different species. And while doing this work, and then we could able to do the specific marker. We could develop the specific marker for Trichoderma genus. And here there was sequences from 6 isolates of Trichoderma virens, and 2 from the NCBA database were taken, and there was a conserved region of 415 base pairs specific to this genus. And through this conserved region, we could develop 248 base pairs marker for the identification of Trichoderma genus. And we have compared this marker with the other genera, and here we could get the amplification of only for the Trichoderma genus, not the other genera. So that's why then we made it an authentic primer for this genus for identification of this Trichoderma genus. Same way, we have also developed the species-specific markers for the Trichoderma and Trichoderma longibrachiatum, then virens, the asperillum, and harisianum. So using the ITS region sequences, we have developed these species-specific markers for all these three. But for harisianum, we have we used RPP2 gene sequences for the development of a species-specific marker. And using these specific markers, we have developed the multiplex PCR for the simultaneous identification of all these 4 genera. And these multiplex PCR products have been eluted and cloned and sequenced and were confirmed through NCBI database that these are these 4 species. And we have also gone for the multi-gene analysis for the identification of the potential, to identify the potential gene sequences for the identification of these different species of Trichoderma. For that purpose, we have done ITS, TEF, calmodulin, beta-tubulin, actin, and RPP2 genes, multiplication and sequencing. And when the dendrogram represents this beta-tubulin region, shows the best differentiation of these species. So under this beta-tubulin, we have also validated using different species of Trichoderma, and then found this is the best region. And through this analysis also, there's and we it was beta-tubulin was the best. And here we have developed this illustrated barcodes for this Trichoderma, different species of this Trichoderma. Then Trichoderma harisianum cryptic species, why we have taken up this work means, but this all Trichoderma harisianum isolates are not biocontrol, having the biocontrol efficacy. So that is why, as per the reports, there were some cryptic species lying in the harisianum complex. So we wanted to see how many cryptic species are available in India. So we have collected some Indian isolates of Trichoderma harisianum and gone for this morphological identification, and there were some overlapping characters also, so that's why we have gone for the molecular identification. So by using both the morphological and molecular identification, we could find there were 5 cryptic species of Trichoderma harisianum. These are the different 5 cryptic species we have identified from the Trichoderma harisianum isolates of India. And we have done the characterization of chetomium, and here we have done the morphological characters and molecular characterization by using the ITS region, and we could establish the different characters of 9 different species of chetomium. Here you can see, and based on the both morphological and molecular identification. And here also for barcode, for selection of the barcode, we have done the multi-gene analysis, and here you can see different regions of this chetomium has been analyzed. And here the ITS was actually gave the better sequence identification, and segregation based on phylogeny is very perfect. But in case of DNA, barcode was high in case of beta-tubulin. So that is why we have concluded that ITS is confirmed as the primary barcode, and the beta-tubulin is selected as a secondary barcode for chetomium species identification, because ITS alone will not be sufficient, and along with the ITS, we have also to do the beta-tubulin sequencing for the proper identification of different species of chetomium. And another area which of a pathogen which we have taken is bipolaris. Here there was a lot of confusion, and there was no common agreement among the mycologists. The same pathogen, like leaf blight, causing leaf blight disease in maize, here in some different scientists have given different names in different years. So that's why I have same Helminthosporium tersicum, Drusilla tersica, Bipolaris tersica, Exorohylum tersicum. So this way there are many diseases were given the same different genera name for the same species. So we wanted to resolve this problem. So for that we have collected the isolate from all other culture collections also, and then done the species identification. So there was a clear-cut differentiation between the Bipolaris genus and Exorohylum genus. Exorohylum have the hilum at the end, and also the black ring at both the the end cells. So, and whereas bipolaris doesn't have this character. So there is a clear-cut difference in the morphology, and that's what also confirmed with the molecular ITS sequences also. And in bipolaris, we could establish the characters, morphological characters for 5 species by using the both morphology and molecular identification. And here also we wanted to select the potential barcode region for species identification. So ITS was selected as a potential barcode region based on this analysis. And the same here also there was another genus, so which is having confusion with the other genera is Bipolaris, Curvularia, and Cochleobolus. Here, if you see, Drusillera australiensis is also named as Bipolaris australiensis, and Pseudocochleobolus australiensis, Cochleobolus australiensis, Curvularia australiensis. The same way for Hawaiiensis species also it is the same thing. And even Specifera also, it was mentioned somewhere in Curvularia Specifera, and somewhere it is Bipolaris Specifera, so that way. So that is why already Bipolaris isolates we have characterized, and we have collected Curvularia isolates from all over the other culture collections also, including our ITCC collections we have taken, and we have established this character by using both the ITS sequences and also the morphology. So morphology, all the time we have taken morphology in the first place. And then only we have compared with the molecular identification too. So that way we have established the characters of different species of this Curvularia. And there is the main confusion is with the 3 species, that is Australiensis, Specifera, and Australiensis, Specifera, and Hawaiiensis. Here the morphology is almost going, but there is a little differences in morphology. And when we have done the phylogenetic analysis of these 3 genera are going with the type genus of Curvularia, not with the Bipolaris. So that's why we have concluded that this Specifera australiensis hawaiiensis species will be with the Curvularia only, not with the Bipolaris. So these will be Curvularia Specifera, Curvularia australiensis, and Curvularia hawaiiensis, not with the Bipolaris. And we have also wanted to select a barcode region for this Curvularia species. For that, we have done the sequence analysis of different regions. And here the barcode gap was more in ITS region. So the ITS region showed high barcode gap, and then ITS can be considered as a primary barcode, better for the Curvularia species identification. And these were the, we have already deposited in the bold also, that is barcode of life database. And we have also done the morphological characterization and molecular characterization of this Phoma species. And we have developed this taxonomical species for 14 species of Phoma. And here we have done the DNA barcoding for this Phoma. Here the ISSR markers as efficient tool to differentiate the variability of genus Phoma. We have used then ITS as a universal barcode, and ITS is as a primary barcode, and then this SSU region is a secondary barcode for the identification of Phoma species, we have concluded. And then Fusarium. We have lots of species and lots of isolates are available with ITCC. So all these isolates, earlier they were identified only based on the morphology, but in morphology there were some overlapping characters. To resolve that, we have taken and did the morphology again, and also along with the molecular we have identified them again, and also we have selected a barcode region for the species identification. Here TEF plus ND6 region. So together have better differentiation of these species. So that's why we have made finalized that TEF plus ND6 region is the best region for the identification of, for making the barcode, and for identification of different species of this Fusarium. And we have also done the morphological molecular characterization of this Rhizotonia betaticola, which is an important pathogen causing diseases in many different crop plants. So here, this is the morphology. We have found different types of sclerotial bodies in this pathogen, and then we have done used the ISSR markers to differentiate this to see the diversity of these isolates, and we could find a correlation between the genetic diversity and also geographical regions. So the regions wise diversity we could find by using these ISSR markers. Then we have also done this characterization in morphological species, Aspergillus species, which is an industrial fungus. And here we have done morphological identification and also the ITS region identification, and we have grouped them into different species. And when we were doing this work, we could find this monomorphic band in case of Aspergillus flavors. So we have eluted that band, and then we have made having 600 base pairs, and we sequenced them, and we have made a primer, 490 base pair primer, is a scar marker for the identification of Aspergillus flavors, and this was validated by using the other Aspergillus species. Here we can see only the Aspergillus flavors has been amplified. And then a DNA barcode also, we wanted to select a potential barcode region. So for that we have done the multi-gene analysis for this Aspergillus also. Here also ITS region has shown the high barcode gap. So the ITS region was found as the best marker for species distribution of Aspergillus. And this ITS region we have also validated using many number of Aspergillus species. Species, these species we have used only the 4 species. We selected this barcode marker, and then we wanted to establish the characters for the Aspergillus. Even Aspergillus also we have many number of species and many number of isolates in our collection. So we wanted to authentically see the establish the morphological characters for our identification purpose. So we have taken many number of isolates and done the morphological studies and also the ITS region studies, and then by combining these 2 identification methods, we have established the morphological characters of 20 different species of Aspergillus. Here you can see all these morphological characters. So by using all these characters, our own studies, we do the identification, whatever comes for the identification in a routine basis, and then these are the 20 different species. We have also wanted to see the chemotaxonomic studies, whether these 20 species are different based on the secondary metabolite production. So when we have done that, many number of species, almost 18 number of species have 1 or 2 chemicals specific to the species, whereas Aspergillus justus and Aspergillus terreus did not show any production of any chemical specific to those species. But the 4 or 5 species have shown the production of pyrrolopyrazine and pyrazine, and these 2 are the antifungal compounds. And 5 species, Terricola, Vistus, Funiculosus, Clavatus, Fischeri, Niger, and Pernicious, Sidovi, and Terreus. These species have shown high amounts of antifungal compounds when we have done this secondary production of different species. And coming to the bioprospecting work which we have done in our laboratory, and we also have many number of trichoderma isolates. We wanted to see whether they have any antifungal activity, bioethical activity against different pathogens. And here we can see this Aspergillus. We have worked with Trichoderma aspergillum, Trichoderma harzianum, and on the secondary metabolite production also we have tried from this biocontrol, whatever the biocontrol isolates we got here, efficient isolates, that secondary metabolite production also we have done. And we also developed the silver nanoparticles from Trichoderma species. And here by subjecting to the 1 millimolar silver nitrate solution. And here after incubation, you can see the change in the color of this solution. Also, when we have gone for the time, we could see these particles, silver nanoparticles, trichoderma mediated particles. And these silver nanoparticles we have tried against Ralstonia solanacearum and Xanthomonas exnopodis. Here we can see these zones, the inhibitory zones. And what cryptic species which we have developed there, identified there in Trichoderma harris. Of the pathogen, that is Rhizoctonia solani. And even Trichoderma afrohorseanum is also better species for biocontrol purpose. And whereas Afrosine and Inhamatum, they are showing the moderate bioefficacy. And whereas the Perify was the least for biocontrol activity against this Rhizoctonia solani. And we have also tried for the, succeeded for development of silver nanoparticles from Beauveria bassiana isolates. And here also the same procedure we have followed. And here you can see the temp pictures of these particles, silver nanoparticles developed, produced by the Beauveria bassiana. And also by Aspergillus niger. Here also you can see this is also tested against the Xanthomonas citri and Xanthomonas solanacearum. And we were successful in getting this inhibition zones in those bacterial plates. And now description of new fungi. We have described 6 new genera and also 11 new species from ITCC. And they were also, these are the new fungi which we have described, and they have been published. And coming to the documentation, which is an important area for 2 sections, HCIO and ITCC. And all the HCIO herbarium specimens has been catalogued and it is available online. And recently we started doing this checklists of different genera. And this is the Puccinia, and we also have this Cercospora. And ITCC catalog and all the list of all these ITCC collection we have in this catalog. And there are also the services, the procedures for sending the cultures and all these regarding the services also, the information is available in this catalog. And this catalog, every 2 years we are revising this catalog, and the latest catalog has been put on the website of IRI. Now it is available online, and the users can use using this URL for the information. And we have also published some training manuals and then also practical manual from our mycology. And this is the digital catalog. Actually, I want to develop this catalog because as you all the speakers have highlighted that traditional mycology is going down. So what I thought is all ITCC cultures, at least we have so many genera here. So all these cultures, these many genera, whatever are available with ITCC, we wanted to develop a digital catalog so that all the researchers or students, they can use these characters and then they can identify there in their own labs itself. Because everybody, all the labs may not be having the facilities of this photography and all. So that is why these authentic cultures which we have in ITCC, and we are making it in fresh cultures and then taking the photographs. And these photographs are taken undisturbed condition directly from the tube, so that the identification will be easier for them to do. So that is why I'm trying to complete this work by next year to take all the digitization of all these genera of available with the ITCC, even at a species level also, the differentiation, so that the end users can use, can do the identification. They need not to send to the ITCC for identification, do in their labs itself by using these characters. So this is my idea of doing this work. And my acknowledgements are due to our director, Joint Director Research, and our head division of plant pathology. Ma'am is always supporting our work, and then she's backbone of our mycology section. And I'm also, my special thanks to Dr. P.N. Chaudhary, Dr. Neeta Mathur, and Diba Kamil for their help, without which I would have not been done this work. And also my thanks to my technical staff and also my supporting staff. Thank you all.
Thank you, Dr. Pramila. Excellent presentation. Excellent. A big applause to you. And you have very nicely highlighted all the subtopics of your title, the conservation, biodiversity, bioprospecting, digitization, barcoding. It was really very nicely organized lecture. I must appreciate that in HCIU, we did all the digitization of the herbaria specimens. And similarly, digital catalog you are having in your mind for all the genera and type species belonging to those genera. It will be really good idea, and you all have images also.
Yeah.
And if it can be really published and it can be even put on the website, it will be really a real science a show window of IRI and also Division of Plant Pathology. So my congratulations to you and your whole team. It's excellent. For all the plant pathogens you are doing, and besides that, you are also doing for the biocontrol agents. Like, it's a very good information. I remember we many times discussed on Trichoderma herzianum, the cryptic species concept. Because normally when we see that there are large number of Trichoderma herzianum bioformulations which are there, given by different universities and different people, but many of them, they are not effective. So we don't know whether they have really recognized the species as herzianum or something, some cryptic species have arisen. So this information will greatly help in correct identification of biocontrol related Trichoderma herzianum, distinguish them from the other Trichoderma herzianum species through in the species complex. Similarly is the case with Aspergilli. So we many times talk about aflatoxigenic and non-aflatoxigenic Aspergilli. So this your barcoding work on Aspergillus species, and also the marker which you have developed for especially flavus, because we say that flavus is aflatoxigenic, although we do have certain markers available, but the work which you have carried out is really of great significance. So, dear colleagues, normally for the presidential address, we do not have any questions. So, we can profusely thank Dr. Pramila Devi for giving this insight into the whole mycology, concising in a very short manner. I would like to, many of the our eminent stalwarts in mycology and pathology are there. So Dr. Manohar Achari can also add on to what you have presented. So I would like to invite Dr. Manohar Achari, sir. Sir, please, we will be delighted to have your remarks and comments. Sir, Dr. Manohar Achari.
Unmute yourself.
Unmute. Sir, please unmute.
Okay. Is it audible?
Yes, sir. Yes, sir. Yes, sir.
I'm very happy to see that Pramila is presented very well, and all the documentation she has shown, and the modern approaches also. And your leadership has helped the Division of Plant Pathology, and especially Mycology Division. And biosystematics it has to take a lead in the years to come. And this is the work that has been done, taking from that of morphology to that of modern era. And it is a kind of evolution that has taken place. And something needs to be done is strengthening of culture center is very important, because we don't have many of the cultures. And madam, that
yes,
in India we have rust pathogens, where there is no culture at all available. and you need to have at least in IRI, because since you are now looking after IRI, and there is a need to develop for obligate parasites also, as a kind of center where living plants can have the inoculum. You know, sorghum itself deposits 20 pathogens. Of course, for plant pathologists, conservation of plant pathogens is not important, because conservation of plant pathogen means it is a negative impact. But nevertheless, if one plant is lost under biodiversity, you know that 30 plant pathogens will be lost. So there may be some good pathogens also. When whole plant is losing, then underneath the ground also plant health is also lost. So soil health is also, many microorganisms we are losing under the rhizosphere region. And some stress has to be given for obligate parasites also develop a kind of restricted, under restricted environment to develop this pathogen culturing and all that, so that in near future for doing, because out of those 10 important top pathogens, Paddyblast is one, Magnaphorte aggressiva, and rusts are also important. And Coltotrichum is also important. Rhizoctonus solani, one of the important soil-borne pathogens. Of course, Dr. Pramila is present, and Rhizoctoni also. I'm very happy. They are doing, HCIO is doing very good job, and Pramila and her team has been doing very good job. And let there be some more cultures to be added. I request all the people who are participating in this, please deposit your cultures in the Division of Plant Pathology, HCIO, strengthen this, and with your new variants and biotypes, ecotypes, and new species and new taxa. And I'm very glad to see the progress that has been made. Thank you.
Thank you, sir. Sir, Dr. Jalali can also give us some inputs. Dr. B.L. Jalali is there, sir.
Please unmute, sir. Sir, you are on mute.
Kindly unmute, sir. Sir, you are muted. Kindly unmute, Dr. Jalali. Sir?
Yes, sir.
Now it's fine, sir.
Well, as I said,
Yes, sir.
I'm not a hardcore mycologist like Professor.
But sir, your guidance.
Nevertheless, I had a keen interest because I have said earlier also, I have taken master's degree in the Department of Mycology and Plant Pathology with a great legend, Professor M.S. Paudi.
Yes,
So that was my great interest in that, and then later on, of course, with my association with the IRI group and Manora Chari and A.K. Sarbai, all other people. The one thing which I was quite impressed was that Pramila and her group, they are making digital catalog. I think that is going to pay in the long run a strong role for future generations using all the techniques of precision in the years to come. Thank you very much. I was quite impressed by the presentation of the material. God bless you.
Thank you, sir. Thank you for your input. Can we now invite the other speaker, or if somebody else can also want to add? Okay.
Hello.
Yes, sir. Please. Sir, I was looking for your name.
No.
Welcome, sir.
Name was not coming. No,
Yes,
name
please.
doesn't have to come.
No, sir. I was thinking
I would only
yes, sir,
like
please,
to
sir.
congratulate
Thank you so much.
Pamela for excellent work which she and her team is doing. And of course, would love to congratulate Rashmi for what has been done. And that reminds me of the day when I was trying to force you in molecular mycology.
Yes.
Years ago.
Yeah.
Okay. So it is so good to see that good work is being done. It has to be continued and, of course, strengthened.
Sure, sir.
That is what is required. So I congratulate Pamela again and wish you all the best.
Thank you so much, sir.
Thank you. Thank you.
Madam Usha.
I just want to say
blessed.
just very few words. And I'm very happy to see the holistic approach what Pramila has taken up. And last 15, 16 years, I am in the department.
Yes.
And with the help of Professor Verma, Dr. Reshmi, DV Singh and all. But then what I have been able to do a lot of work with them, along with them. And also I observe now the entire group, biology group, has evolved, very much with the multidisciplinary aspects of the thinking has come. Initially, I didn't find really that much of the multidisciplinary aspects to be taught. But now after seeing Pramila's work, and then I also see Rashmi's work and all the students, Prakash, everybody is known to be, I keep discussing with everybody, then everyone has evolved into a different type of thinking, that is a holistic approach and a molecular approach, as well as a taxonomical and application oriented. The most important, as Manoharachari was telling, that whatever we make, that has to have some application value. So it also has entered into a patenting era. So people started doing towards that also. That I'm very happy and hearty congratulations to Pramila and Diva and all youngsters, Prakash and Mugam, and all is responsible, main responsibilities with Rashmi, as she was supporting them. And also encouraging people like Professor Verma is there, and then there are many. Jalali sir is always with them. And I am very happy to be associated with this group, and very thankful also to all of you for, making me a part of that group for the thing. Many more achievements for you, Pramila, with your dedicated efforts. I am very sure that all your objectives you will be able to achieve for that. Thank you very much.
Thank you, ma'am. I will add a few words to it that Dr. Pramila also had made efforts earlier, sir. All of you remember we were having mushroom training program also, and we were supplying many years ago. We were giving the spawn. We were supplying the spawn. Now Pramila had made efforts to get a NABARD project for again revising and revisiting that spawn production. And it's a 2-year project, and maybe we will again initiate the spawn preparation and supply your spawn. So with her initiative, we could develop that project, and it has already been sanctioned by NABARD. So that's another addition to my ecology section, sir. So with all your wishes, and I'm so much thankful to Professor Verma. He always, when they had a UNDP project, this Advanced Center of Plant Virology, sir always said, "Tum theek karo, tum apna molecular karo." I remember. So slowly we ventured into, and now with morphology and molecular together, our mycology and fungal pathology group is doing good, sir. We are quite satisfied, but really we have to go much ahead. So thank you so much, sir, for all your supports, Dr. Professor Verma, Dr. Jalali, Dr. Manoharachari. All of you have given us so much of boosting and support that we are thankless on this. Thank you so much. So, sir, with your permission, we will proceed to the next speaker. Ji, sir. Ji, sir.
A word before you end up this lecture is,
Ji, sir.
We do apologies again.
Ji, sir.
Since you are the custodian at the central office also, as a joint director,
Ji, sir.
and also as head of the division, can you retrieve the word mycology in your team?
Sir, 2-3 places I have already spoken
no,
to.
Sir,
with your support, and I don't know, I will try, sir. Definitely.
Okay, thank you.
Now with our very dynamic honorable director with us, so we will just look into it, sir, how we can.
Well,
and I think he will have all the support. That I'm very sure that he's so good, so humble. And
well, it's a debatable issue.
We can only.
I am responsible for getting it removed. Okay, so and I'm happy to debate. I've debated with many, including I'm sure with Jalali. Okay.
So
Sir, निकल गया.
That is why I wanted to raise the
I
issue.
know you are saying it deliberately.
No.
1988.
Please.
Okay.
Not that way.
Okay, sir.
Like
No
a-
worries, sir.
like a-
Everything is good, whatever is there or whatever can happen. Thank you so much, sir.
Sure,
So we
wish you
will
all the
proceed.
best.
Thank you. So I invite G. Prakash, Dr. G. Prakash, a budding scientist in the division. First of all, my congratulations to Dr. G. Prakash, who got M.K. Patel Young Scientist Award by Indian Phytopathological Society during the year 2019. And now he is going to present this award lecture. G. Prakash is rice pathologist and very efficient. I must say that he's trying his level best to come up and doing good job. He is associated with me in a CRP genomics project, and he's doing on Megnaporthe species, the genomics, and the trying to do multi-locus sequence typing, and also recently he got one NASF project also on maybe on transgenic rice, one as co-PI and another one on genome editing. So he's working extensively on blast pathogen on rice. So he's a very good team player with our director, Dr. Ashok Kumar Singh, as a rice breeder, and he as a pathologist associated for blast. So I invite Dr. G. Prakash.
Yes, ma'am.
For
Thank
his
you.
lecture on understanding the pathotypic diversity and genomics of Megnaporthe for management of blast disease. So G. Prakash, over to you.
Thank you, ma'am. Thank you very much for your kind words. Ma'am, is it visible, ma'am?
Yes, Prakash.
Yes,
Yeah.
Prakash.
Thank you, ma'am.
Yes.
Thank you very much. Good afternoon to all of you. Yeah, today I'll be talking on understanding the pathotypic diversity and genomics of Megnaporthe for management of blast disease. The first part is obviously I'll be discussing on pathotypic diversity and followed by genomics of Megnaporthe. The Megnaporthe caused by blast is one of the top 10 fungal diseases that threatens the global food security. And it infects the huge number of agriculturally important cereal crops, starting from rice, wheat, oats, barley, and millets, followed by a lot of grass species. The sexual reproduction has been reported from South Asian countries, but the majority of their species means majority of their evaluation, it's happening through clonal propagation. So that's why the management of blast pathogen is often challenging. If you take our Indian scenario, the blast is very serious in case of rice as well as millets. So understanding the population structure is essential. Population structure or virulence structure or pathotypic diversity is a key essential for devising any kind of management strategy. So we were focused on establishing the isolates for pathotyping work. Yeah, this table depicts the area where the state where we established the isolates and the number of locations and how many isolates we have established. From Basmati regions, we could establish 50 isolates. From non-Basmati, we established 57 isolates from the different states. So those who are working in blast, they would have understand the difficulty in isolation the pathogen because of the infected blast lesions, which always closely associated with the other saprophytic fungus, that is Carvularia and Alternaria is major saprophytic fungus, which always associated with the infected blast samples. So when you go for direct host plant transplantation, host tissue transplant based isolate, most of the time you won't get success. Maybe in fresh samples you will get you can able to isolate, but once the samples become old, it's very difficult to get the isolation. And moreover, the sporulation issue also is a very key important one. So initially we thought to address this issue. For that we established the isolation protocols, that is spore drop method or lesion print method. In this method, there is no direct contact between the infected samples as well as the media. It is the indirect contact between the infected tissue as well as media. Because of magnopathy tendency to move towards moisture, we could able to get the single pore colonies in case of in the media. So by using this method, we could successfully establish the wide number of 107 isolates from Basmati as well as non-Basmati representatives for our pathotyping, host-based pathotyping work. So we know more than 100 blast-resistant genes has been reported in different rice genotypes. Among them, around 26 genotypes, means 26 origins, resistant genes has been cloned and characterized. So in our study, we have used 25 resistant genes for host-based pathotyping work. So these 25 origins has been in LTH background, that is Japonica background, 24 monogenic lines. It is in differentials, and one gene, this PA54, is in Pusa Basmati 1 background. So for the TETUP also we have taken as a, TETUP is one of the dominant resistant source for magnopathy, blast pathogen. So we have taken TETUP also. So this is how we do the phenotyping. Initially, we raise the seedlings. Once the seedlings reach the 3-leaf stage, parallelly we multiply the different isolates where we inoculate, and we keep the phenotyping facility conditions like temperature and high level of humidity and the light as well as darkness. Darkness, actually, the pathogen loves dark period of, we need a lengthy dark period. Then after 7 days of inoculation, we'll go for all kind of observations based on different scales, 0 to 5 scales or 0 to 9 scale, like that. So here you can see the diversity of magnopathy population from Basmati regions and non-Basmati regions, the number of isolates, how many we isolated, and number of genes which has been defeated, which can able to knock down. So based on the percent virulence pattern on different monogeny differences, we could categorize the isolates into highly virulent and least virulent category. 21 isolates and highly virulent, and 55 isolates were virulent, and 31 were less virulent. So this table depicts the virulence frequency percentage on the different genes, monogenic differentials of Basmati rice region, starting from Western Uttar Pradesh, Haryana, Uttarakhand foothills, and Delhi foothills of Himachal Pradesh and Punjab. Tested 50 isolates we have tested. The overall output of this this one is we could able to see identify some of the effective genes for Basmati growing regions, that is PA54, PETA2, PETA, and PA9, PAZT, PAZ5, PA12, and PAZ. These genes are very effective for Basmati rice growing regions. Similar way for non-Basmati regions, we got the level of genes, effective genes is different from the Basmati one. But even though it is more or less similar pattern, but we found the effectiveness is different from Basmati as well as non-Basmati. Here we found PA12, PA9, PETA2, PA54, PAZ5, PA20, PAZ, PETA, and PAZ5. These genes were effective. When we depicted these in state-wise as different genes for even for deployment purpose, here we could see all different genes has been deployed for different states. For example, in UP, you can see that these genes, but in case of non-Basmati, if you take Maharashtra or Karnataka, the genes might be different. So this how we deployed the effective genes for different state purpose, different state. Then further, overall, the effective genes, if you based on all the isolates against the monogenic differentials, the high level of resistance we are obtained from PETA2. Around 67% resistance spectrum of 107 isolates what we performed. Then PA9 followed by PA9, PA12, PA54, PAZ, PETA, PAZ as well as PAZT. These genes, which is major genes, which is having kind of resistance spectrum against the existing population of the Magnopothe. So these genes can be individually or in combination can be used for gene deployment strategy for effective management of BLOS disease. So then further we categorized into different races by using international race classification system. Here we could see that I listed the highly virulent races from the different states, and 107 sample. This is on the reaction on the monogenic differential, the cataloging of race flora, which we did by the phenotyping on the on monogenic differentials. So I told already TETAP is one of the dominant resistant source for BLOS pathogen. In our collection, we had some of the compatible, even TETAP compatible isolates also. Few isolates I'll tell from one isolate from Cuttack and Punjab, and from Haryana and from UP also we had some. This is some more isolates also we had in our collection, which is capable of knocking out the TETAP. So it is compatible with all the existing resistant gene which we handled. So these information, the information which we generated, it is not only given the effective genes, not only it is depicted the effective genes, but also it is helpful used for to identify the diagnostic isolates. That is very key important for phenotyping of resistant varieties. Diagnostic isolates means, for example, the gene PI9 introgressed line, which we have to evaluate those line with the P9 virulent isolate as well as P9 avirulent isolate also. So both the combination we should have, a virulent isolate also we should have, and the avirulent. That has to given only infection to the P9 background only. So any from breeder side they will be having the marker based identification, but even biologically we have to validate by using the diagnostic isolates. So likewise we have a different major gene based diagnostic isolates in our collection, which helps in developing these resistant varieties. We were associated with developing these different resistant varieties in with different gene combination. This is PAZ5 PA54 combination as well as PA9 alone itself given resistance in PUSA 1637. Likewise, recently last year 2018 it is released PUSA 1850. This is BPT5204 babrand. This is triple G gene combination. So we had a diagnostic isolates to do the thorough phenotyping, so that we could able to achieve this this achievement. So my second part of the talk is which Madam already told, Magnopothe genome sequencing, where Madam is Dr. Rashmi Agarwal is a PA of that project. The purpose of this genome sequencing, Magnopothe genome sequencing, we would like to compare with our Indian strains where it is standing when you compare with the world level strains, as well as their genome level variation. What kind of genome, how much level of variation is there? Whether those information can be useful to study their pathogenic means what you call plant pathogen genetic analysis, speciation, as well as host specificity, and to track the pathogen movement, all kind of things. We keep in mind, we addressed one of the strain of rice BLOS strain, that is RMG-DL. The key feature of the strain is it not only infects rice, it infects wheat, oats, barley also. So it is one of these highly virulent strain. In the background of we isolated from the host is from Sorna background. So which we sequencing was performed. The high quality genomic DNA which we have isolated from by CTAP method, then we have used the genome sequencing by hybrid approach. For short read, we used a HiSeq 2500 by using chemistry of 2x125 base pair. Then for long read, we have used PacBio P664 chemistry and the standard bioinformatic pipeline which we have used for all further analysis. Yeah, overall in assembly we found the 996 scaffolds, which is of genome size of 34.8 MB. which of average scaffold is 45,894 base pairs. The number of genes is 1274 based on the gene prediction model, Augustus model. This is the gene assembly statistics we received by sequencing of RMGDL. The other strain which infects pearl millet, the passport data is here. It is isolated. It is from New Delhi experimental farm and non-infectable. For this strain also is capable of infecting wheat, oat, barley, other than their own host, but other host. So this for the house information, this is our first draft genome of pearl millet blast throughout the world. So this we could success with the help of our team members of CRP Genomics. Yeah, same strategy we used here. We used a hybrid approach. Illumina NextSec 500 2 into 150 base pair chemistry as well as PacBio RS2. Similar way standard in bioinformatic pipelines we followed. Then the overall data what we received by parent we received around 13.1 GB. In mate pair we received 3.1 and PacBio 1.1 GB. When we then these reads assembled into scaffolds by using Spades assembler, genome assembler. Here we received 341 scaffolds with a genome size of 47.9 MB, which average scaffold of 765,468 base pairs. The number of genes in Magnopothe gracia as a prediction model. So these two genomes, both the genomes which we have compared, which we have in silico comparison we made for RMGDL as well as PMGDL. The statistical data is here. Genome size I already mentioned 48.89 MB as well as 34.81 MB in case of PMGDL as well as RMGDL. The gene calling resulted 10,218 genes as well as 9,748 genes. Our key interest on this is virulence factors, because virulence factors plays major role in host pathogen interaction, and our beyond that our interest to study their host specificity, which I'll come later. So yeah, this both the genomes which we compared with other existing genomes, the average nucleotide identity between these two genomes is 83%. The difference between these two genomes is 17%. Average nucleotide identity differs. When we compared with Magnopothe poye, it's a kind of root infecting blast fungus. We found the differences around 17% in case of Gracie and 29% in case of Varaceae. We found. Then further we analyzed the protein prediction by interproscan analysis to see the protein family and superfamily as well as conserved domain regions. Here we have compared with the the reference genome. The reference genome is Varaceae, Pyroglyria Varaceae 1715 is the reference genome. When we compared our genome with the reference genome, we found the common as well as uncommon as well as shared predictor proteins. Similarly, we compared the caseins also, carbohydrate active enzymes. Here you could see the unique as well as common as well as shared carbohydrate active enzymes. Our key interest, which I have already mentioned, is virulence factors. The key the virulence factors in case of Gracia, that is PMGDL, is two virulence factors, which is very unique in case of Gracia. Similarly, 4 virulence factors, which is very much unique in case of Varaceae. So further, we thought to utilize these in silico analysis into for wet lab validation, which I'll come later. We know this genomic information, what we generated, we addressed a lot of biological questions by using the genomic information. The one is we study the population genetic analysis by using multi-locus sequence approaching, selecting 7 effector genes and 11 housekeeping genes. And both the population we studied. And further, we developed a means pathogen-specific marker for diagnosis purpose, which these two parts I'm not going to address in this presentation, because our key, we were very much interested in this area, that is on host specificity. When we do the phenotypic, comparative phenotypic, we found these kind of observations. For example, rice isolates when we inoculate on other host, pearl millet and finger millet, which gives infection only in case of rice. So that what that indicates, even though blast pathogen infect a variety of hosts, different hosts, but each pathogen they exist in a specific form. So to address this one, the in silico analysis of whatever we compared both the genomes of rice as well as pearl millet, we taken some of the hypothetical genes of rice blast pathogen. Then we proceed of their expression, whether the in silico data, whether it is exactly expressing in case of when you do it in biological material. So we studied the expression analysis of this hypothetical Magnoporthe genes using qPCR analysis. This is experimental design where we used two susceptible variety and one resistant rice variety, BPD5204 PB1. This is Pusa Samba 1850 is a resistant one, which is having 3 major genes. Similarly, pearl millet one line for comparison purpose we have taken. The respective inoculation, Magnoporthe rice inoculated on rice, grace inoculated on pearl millet. And we collected samples on different time period, starting from 4 hours to 120 hours, up to the symptom expression level. Then we isolated the RNA and cDNA synthesis, followed by qPCR for gene expression studies. Yeah, these are these 3 genes or the hypothetical genes which we have taken for this study, and the 40S ribosomal subunit as a normalizer or endogenous control. The experimental details is here, real-time experimental details is here. Yeah, the first gene, that is 3349, we got the results like this. We thought it's specific for MRSA, rice blast pathogen, but the results are a bit controversy. We could see the expression in both pathogen, pearl millet as well as pearl millet as well as rice blast pathogen. But the rice blast pathogen, similarly in case of host also, when you interact with host, we could see expression in case of rice and all the 3 rice varieties and pearl millet variety also. This is pearl millet variety also. In this is a resistant one. The expression level is very low. It is well understood that it's the line is resistant one. Although expression in both pathogen it happened, but the level of expression, the fold change is high in case of rice blast pathogen. The next gene which we have taken is another hypothetical gene, that is 5524, the gene ID. Here also we received a similar kind of result. It is expressed in both the pathogen, but the level of fold change is high in case of rice blast. And interestingly here, in resistant varieties also we could see the high level of expression for expression, which indicates maybe this gene or this gene may a novel effector which we don't know, which we yet functionally we have to validate those genes. Then only we'll come to know this, whether this gene, how much potential of this gene and what is their role. The third gene is 8852, which is another hypothetical gene. Here, very interestingly, we got this gene expressed only in case of rice blast fungus, which it is not expressed in pearl millet. Similarly, it is expressed during interaction with the host. It is expressed with the susceptible to host, BPD5204 and PB1. This it is not expressed in case of pearl millet during interaction as well as pearl millet fungus. So it is very clear that this gene is having a potential to play a role of pathogen determinant in case of host specificity, which we have to explore further. So this gene, we further planning for functional studies to establish its role in case of their host specificity. So that's it. That's it for now. That's it. My presentation. I would like to summarize the presentation. We could establish the simple and efficient methodology for isolation. Because this method recently we tried even 14 years samples, 14 years, 2006 collected samples, still I remember, we could able to establish isolate now also. It has been stored in -20 with a live sample, which now we could able to establish. The sound, very simple method, but it is very much effective for isolation of especially four-leaf fungus. I never tried on other fungus, but it on my experience, it may work on other four-leaf fungus also. So we could develop host-based pathotyping for to identify the dominant effective genes for effective management, and we could identify the highly virulent races from different rice growing regions, and we associated with the blast resistant varieties. We sequenced the first genome sequence of pulmonary blast, followed by Magnopothe genome rice blast also, and we analyzed the genome, both the genomes, and we found specific virulent factors and carbohydrate active enzyme. And some cases we found unique pathways. Compared to pearl millet genome, blast genome, the rice genome size is very low. So it is very less in adopted one. So then we identify the host pathogens also, which functionally have to be validated. So I thank my mentor or chairperson, Dr. Yudi Singh, for his guideship, as well as still now he supports me in various manner. And my sincere thanks to Dr. Kumar. He made a significant contribution in genomics part, and also my collaborators from Rice Genetics for their constant support, Dr. A.K. Singh, Dr. Gopalakrishnan, and Ranjit, and Rajshekhar. He's my colleague. We work together here. He has supported a lot in during my PhD program, as well as further my work. Still, we have a very good rapport in research. So my acknowledgement to funding agency, CRP Genomics, as well as NHP CAST. Their support is highly acknowledged. And we recently, Madam told already, we are in future line of work with genome editing for enhancing blast resistance. And my sincere thanks to head division of plant pathology, Dr. Rashmi ma'am. Even she is a PA of CRP Genomics, and he supports a lot for encouraging the work, as well as all kind of activities. Thank you very much all.
Thank you, Dr. Prakash, for excellent presentation. And I know that you are working very hard to maintain the cultures, do phenotyping. I have personally visited all your facilities, and it's your really dedicated hard work, which phenotyping is very important, and it's very difficult also, especially for the blast fungus in rice. So thank you, G. Prakash, and once again, congratulations. And very nicely, you have summarized the whole work, and at a very young age. So we look forward for a much more work out of you, and very good project you got on genome editing. So it's a new line of work, and you're the first one to get in fungal pathology in the division. Of course, we have one in virology, one or two maybe in virology. Dr. Anirban in virology is taking up. So thank you.
Thank you very much, ma'am.
Sir, we are already behind schedule by 1 hour. I am told that we have to quickly proceed to the next session. So any quick support, comments, encouraging?
If madam, if you
yes, sir,
allow,
Dr. Dubey. Yes, please, Dr. Dubey.
First of all, I would like to congratulate Dr. Prakash for doing excellent work and winning this award, Dr. M.K. Patel Young Scientist Award. My question is, he has tried to make a deployment of the varieties having the different resistant gene in different area, just like the wheat, where the success story has been noticed. But the problem in the blast is that we don't have the phenotypic evolution of the races in that particular area. Unless we don't have the information of evolution of the phenotype races of a particular area, we cannot deploy any kind of the variety having the resistance. Simply what he did, collected the isolate from basmati and non-basmati and identified screened them against the different cultivars or germplasm or lines, maybe the near isogenic lines having the resistant gene for a particular isolates. This will not serve the purpose. First of all, he has to find out the races prevalent in that particular area. Then this study has to be done. Otherwise, it appears to the wrong way in the reverse side. It has to come the forward side. As second one, the hypothetical gene for the host specificity. To do better, you have got the effectors, 2 effectors and 4 effectors, which is entirely different in both the isolates. So you have to look for that. You have to do the wet experimentation for these 2 effectors, characterize them, isolate them, and let us see what effector is there, which gene is governing these effectors, and you have to try to clone them and sequence them and identify them and do the validation of these. That better rather than doing the blanketly for the hypothetical gene, which is a long process, and maybe it will not be work later.
Prakash, I think these 2 which we have taken, these are hypothetical specific only.
Yeah, yes,
isn't
ma'am.
it?
It is
These
hypothetical
are 2
specific.
because they were not identified, but they were unique. So those only you have selected.
It is on in silico we identify.
In silico,
sir.
the same
Further,
he has
it has to
taken.
be wet lab.
He
We
has
have
taken
to
the same.
Whatever in silico.
But not the sequence, the whole gene isolation and sequence work he has not done, only expression analysis.
Expression I studied, sir. Now I narrow down into some few one gene now we found. So further it has been we can explore further their functionality, how it is working and all kind of things.
Another suggestion for the cross pathogenicity, what you did the pearl millet and the sorghum with the blast, you have to check with the different variable environmental conditions. Don't keep in the same because this blast pathogen can go on the pearl millet also, but maybe due to change into the environmental condition, you have to modulate and change the different environmental condition, humidity and temperature. And let us
so
look.
We carried out that one also. So we did.
They did.
Okay, thank you very much.
Okay, well taken your suggestions, and also think about monogenic differentials which you have. I know you have the answer and you have done extensively, but you can separately discuss with Dr. Dubey. Yeah.
Okay, sir. Thank you, sir. Thank you very much for your suggestions.
You take suggestions and you should do whatever. If you have not done, you should do that.
Sure, ma'am. Sure.
Thank you, all of you.
Thank you, madam, for it.
I just want to congratulate.
Madam Usha.
I just want to congratulate Prakash for his very hard work and a systematic work which he has been doing, and with the new suggestions of Dr. Dubey and also with Dr. Reshmi, I am sure that you are going to achieve your objectives very well. And keep up the spirit continuously, and wish you all the best for your good research work. That's what I just.
Thank you, ma'am. Thank you. You are a great inspiration to everyone.
Thank you so much to all.
Thanks a lot, ma'am. Thanks a lot.
Thank you.
So we profusely thank both the speakers and all the audience, all the participants who patiently listen to both the very prestigious award lectures. I must say that is also a kind of award only, the presidential address and also the address by Dr. G. Prakash. So I think we should end here. Thank you, all of you. So I think I'll give it to the floor. I'll give to, thank you all, both the reporters who helped, who must have taken the notes also. And I thank Dr. Dinesh Singh also. So I think at present he might have left because he had another training program going on where he had one lecture.
Yeah,
so I'm thankful to Dr. Dinesh Singh and both the reporters, Dr. Malkhan Gurjar and Dr. Amrita. So, and I'm thankful to the organizers for giving me this opportunity, IPS and President IPS, Zonal President IPS and Diba. Thank you all of you, and
thank you so much, ma'am.
to Amrita. Yes.
Thank you, ma'am, for your valuable remarks. Now we're having the important session on the keynote lectures. I now request Dr. B.L. Jalali as chairperson and Dr. A.C. Dubey as co-chairperson to conduct the session. And the reporters are Dr. Vishnu and Dr. Pardeep Kumar. So, Jalali sir. Hello.
Unmute, sir.
Unmute kar
Kindly unmute yourself, sir.
Well, welcome to this second session, which is supposed to be two very important keynote lectures. I hope both the lecturers are available.
Sir, available, sir.
Professor Manarachari is there. What about the second speaker?
Hello, sir. I am here.
Shilpi
I
Sharma is
can
also.
see you. Anyway, let us start with the first lecture. Professor Manarachari, and the title of his talk is Biodiversity and Taxonomy of Fungi, an Indian Perspective. I wonder if at all Professor needs an introduction. He is already an icon in the domain of mycology at the national and international level. Nevertheless, for the convenience of the youngsters, I wish to highlight Professor Manohar Acharya, who has served Osmania University virtually for about half a century, if I recall correctly.
Yes, sir.
Close to half a century in different capacities, and also the Vice Chancellor of Central University, Indore. All through his career, he has guided 50 students for PhD and published more than 600 research papers and 30 books, including 5 books by the Springer. In the domain of mycology, he has described 20 new genera and 120 new species of fungi, and he had very distinguished postdoctoral research stint at UK and Germany and USA. Where has he gone? Hello? Yes. And got a DSc degree from the Mysore University in the year 1999. Recipient of 5 national awards, including the E.K. Janaki Amal National Award by the Ministry of Environment, and 5 Lifetime Achievement Awards. 5 Lifetime Achievement Awards from government, and also award from the government of Canada. He is also the fellow of the 2 prestigious academies, the fellow of the National Academy of Science India and also the National Academy of Agricultural Sciences, and also besides being the fellow of the IPS and IBS and IMS. And his contributions in nutshell are immense, particularly in the domain of mycology, biodiversity, taxonomy, conservation, biotechnology of microbes, fungi, and currently he is serving as the honorary Nazi senior scientist. What has happened?
Slides.
So with this, it is always fascinating to see him delivering lectures and giving us some newer insights into the domain of microbes, particularly fungi, in different situations. With this, I invite Professor to deliver the keynote lecture on biodiversity and taxonomy of fungi.
Thank you, sir. I am grateful to Professor Jalali and IPS of Delhi Zone, and then Dr. Rashmi Agarwal and all other people who are there, and Dr. A.C. Dubey and the rapporteurs. I thank them I thank Professor Jalali sir for his encouragement and valuable help that he has given to me from time to time, and also the being the chairman of this particular session. He has introduced this tiny fungus like a giant mushroom, which I may not deserve much. Coming to this second slide, please. You know that most of the youngsters should know that fungi are eukaryotic and heterotrophic. They are osmotropic and they are holocarpic to eucarpic. And you know the Professor Jalali has said that there are the holocarpic fungi being not worked out. Only Dr. Pavgi was responsible for discovering this, and later on Ramdayal has taken over. And there is a lot of work to be done on holocarpic chytrids, and which has been neglected in this particular country. And then most of the oomycetes, they have got cellulose, and chitin cell wall is the major component in many of the fungi. And most of the scientists, neither botanists nor microbiologists nor animal scientists, consider this particular one as their own kingdom. Therefore, fungi created their own niche, and therefore now it is included as a separate kingdom. And then they reproduce by means of asexual and sexual spores. Uh, how old are these fungi is a very difficult question to answer, but based on DNA sequence, the fungi might evolve between 1.06 billion years to and 760 million years ago. And we have very poor fossil record. And fungi are everywhere. They are cosmopolitan distribution. They are ubiquitous. They are there on the living vascular plants as obligate parasites, as saprophytes on the litter, and also as biotrophs, and also in the soil and root and soil-borne and root-borne diseases are caused. And non-vascular plants also like cryptogams, they occur in the case of algae also. The chytrids will parasitize, and bryophytes and pteridophytes, they are there. In the plant exudates and also on the grass droplets also you do find fungi, and most of the saprophytic fungi are there on dead plant pods, and other microbes and analogs they are present. And on vertebrates also they are present. Most of the skin disease causing fungi, they belong to mycota. And then non-vertebrates like echinoderms, they get infected by hypochytrids, and then so on and so forth. And then in water, air, soil, and then keratinophilic substances and coprophilous and aeroaquatic, between the water and the air, there is a space which is occupied, empty space is there, that is occupied by aeroaquatic fungi. And water droplets and rhizosphere, phylloplane, rhizoplane, compost and extreme habitats, etc. Next, please.
We have one more lecture also. Obi, she is also having a meeting at 1:30.
Yeah.
Dr. Pramila, please mute.
Yeah, why should fungal diversity is a variety of life on Earth, and the genes through their organismal and ecological lens, and you know that their ecological levels, they differ, and they are ubiquitous and cosmopolitan. They differ in their morphology, reproduction, ecological behavior, biochemistry, physiology between species, and so on and so forth. Now, next, please. I need not say, you please carry on. Why should we estimate this fungal diversity? Fungi are second species rich organisms occurring after insects, and it is a major challenge to complete the global inventory as compared to other organisms such as plants. And fungi play a very key role in the oxygenation of earth and a significant role in the ecosystem dynamism as decomposers. And there are a number of fungi whose individual role needs to be understood, and this is still unknown. And then Hawksworth 1991 gave an estimate of 1.5 billion fungi. This is the world estimate. And later Hawksworth and Lucking in 2017, they have given up to 3.8 million fungi. Recently, based on morphological studies and high throughput sequencing, Wu et al., they have said that it is around 13 million or 14 million fungi. Now it is clear that there is a hidden wealth of fungi which is underneath the different ecological niches, including micro niches. A total of 120,000 fungal species have been reported from all over the world, which belongs to around 6,000 genera. And then in India, we have reported is estimated around 5 to 10 lakhs of fungi. However, as on today, we have only 29,000 fungal species have been identified. And this remaining, which belong to 2005 genera, and the above data clearly indicates that there is a need to take up a lot of field work in such a fungi occurring under micro habitats, both in the world and as well as in India. And scientific necessity is ours because bioprospecting of fungi is an important aspect. Therefore, we have to go for this particular group of fungi which are occurring in different ecological niches. Then you see, one has to plan how one can do this particular one, planning and implementation of all this taxon biodiversity. First of all, you observe, you go for a specialized ecological niches and the litter and all that, and then collect the materials and detect the fungi first at the field site and come to the laboratory and do all the observations, and then data capture and rationalization, characterization, then output. And then these are the conservation methods which has to be taken into consideration, ex situ and in situ conservation, because fungal wealth is very important, and lyophilization, mineral storage, water storage, soil storage, and so on and so forth, like silica gel storage, deposition of cultures at national and international culture collection, that is very important, and preservation of fungal herbarium is very important. And we have, these are the centers which are there, and I will come to this Indian type culture collection. Now they might increase the cultures and around 3,800, but it may be more now. And only 7% of fungi have been cultured in India and elsewhere, and there is a need to use conventional and non-conventional methods besides some innovative methods which are essential to culture these fungi. And NBM is only concerned with agriculturally important microorganisms. In India, we have different climatic conditions, forest types, soil types, and it is a must to have a rich wealth of fungi, and different climates of various countries, also various continents is here. And you look at the world estimate of fungi, it may take more than 10,000 years to study fungal wealth. From the fungal estimate of India, as mentioned above, it may take around 5,000 years to study these fungi. Unfortunately, many of the taxonomies of the world and also in India have expired, and some of them have become old, and available taxonomies are very few and endangered. So this is the picture of the taxonomies and role of mycologists in India, 1907 to 2000. Many of the noted mycology like Butler, Mundkar, Saxena, Tandon, Tirmalachar, Dasgupta, Bilgrami, Kapoor, Bhargava, Mukherjee, Agarwal, and Paugi, Mehrotra, Kamal, Agnihotrudu, Niranjan, and then Munjal, Lodha, Lakanpal, and all others contributed immensely for the Agnihotrudu. They have contributed for Natrajan, they have contributed for the growth of mycology and described many new genera and species. However, 2012 onwards, we don't have much of this particular genera and species. The rules stipulated by national biodiversity does not allow the deposition of material outside India, CBD also, and nor mycologists of other country can deposit. This has become stumbling block and hitch to deposit neither genera and species. Therefore, only new genera and new species have been published in India, and they need to be, because they are depositing in other countries and they are publishing, and thus it is clear that existing mycologists have to bring this sad state of scientific matter to the notice of National Biodiversity Board and CBD. Any of the difficulties that mycologists are facing, they are approaching foreign mycologists. Next one, please. Recently, Chinese mycologists came to India. I don't know how they have come, with whose permission, and gone to the forest of Meghalaya along with some mycologists of some institute, and they have discovered a green light emitting mushroom, Rhodidomyces, which is a bioluminescent and very important fungus. Therefore, it is essential that NBB and CBD have to relax the rules so that our diverse estimate and discoveries of new taxa will go up, and respectful position India will have in the world. So taxonomy, the science of classification, naming and describing the organisms, all of you know this, evolutionary and phylogenetic relations between the organisms. Next one, please. Then this is the fungal taxonomy, morphological data, vegetative reproductive structures, microscopic and macroscopic, ultramicroscopic, cell wall composition, chemical profiling, scanning, electron microscopy, and then we have immunological methods, and then enzyme assay, carbohydrate assay, protein profiles, molecular techniques, all these PCR, RR, DNA barcoding, carbon is the source, and based upon glycerol, orbital, mannitol, and other such kind of enzymes. Many of the yeasts have been very much neglected. Secondary metabolites are very good diagnostic features like polyketides, glycopholin for penicillium, Taxol for the taxomyces. Next one, please. Multi-billion-dollar industry has been developed because of the Taxol. And then this is the hierarchy of the taxonomic classification that everybody knows. Next. Then this is the recent classification Septora, where the Eumycota, Basidiomycota, Ascomycota, Zygomycota, Chytridiomycota, very simple classification. And the Heterocantae, that is 2 flagellae. And then you have got Oomycetes. Go to the back, please, go to the back. Oomycetes and Oomycetes, now scientists, mycologists are thinking that it has to be raised to a level of kingdom, and Hypochytridiomycetes and Myxomycetes earlier, which was in fungi, now it has been included under protozoa and Plasmodiphora mycota also. Next. This is the Hibet classification based upon DNA sequence and all that, and these are some of the classes. And then this is recently another 2019, another classification has come, and you can see that they are narrowed down the class, they are narrowed down the species and genera, and then because of the peculiarities that are existing, now these are 16 or 17, maybe more, and that they have classified the fungi like this, and that is different phyla have been created for each group of organisms. Some important observations are yeasts are neglected in our country. There might be 200 species of yeast which are very important for industry and present in oceans. Extremophilic fungi occurring in deserts and thermophilic springs and thermophilic habitats, and the habitats of adverse conditions are very important biotechnologically, hence needs to be studied. Not much data is available. Mangrove fungi and water fungi have been neglected, and they are very much important as they have got a lot of secondary metabolites. And study of chytrids has been neglected in India. Most of the chytrids are parasites and algae, and some of them occur on echinoderms, insect gut fungi. Insect gut is also very much neglected, and therefore it is very important to know all these groups of fungi which have been Lebalbinemycetes has been neglected. It does not mean that other fungi are neglected. A lot of literature is available on other groups of fungi. There is still hidden wealth of all other fungi also, which needs to be our attention in view of their economic importance. Recently, people working on different habitats, they have searched for new fungi. They have come out very industrially important. ICN has given the data on fungi. In the world, ICN has reported 1,512 fungal species. In the year 2016, they are declining species. But in India, we don't have any kind of data at all on Red Data Book. Dr. N.K. Harsh of Forest Research Institute, Dehradun, has been assigned by the Minister of Environment to go into this particular one. And whereas a country like Bulgaria has got ICN red list, and we don't have this one. So from the available data, the Tibet and Nepal and India, Ophio, and we have got Ophiocordyceps sinensis. This is Desi Vigara and also is useful anti-cancer drug, is well-known fungus, and it has become endangered. It needs to be cultivated under laboratory conditions. This is the largest truffle. These are the variety of the fungi, Allomyces, Aplignia, Pythium. Nobody has asked Pythium to release the juice pores into the vesicle. It is a characteristic feature. Biodiversity is very high, and morphologically the fungi are different. It is only we differentiate, we are not able to differentiate them and identify them correctly. Our naked eye, scientific naked eye need to be very vigilant. Next one, please. Now, these are all fungi which are very important, but I would like to mention Dr. Saxena, SB Saxena, he has discovered Saxena vasiformis, a soil fungus which has become unique indigenous fungus, and it has needs special attention because it has become parasite on the people who have lost their legs and limbs and other things. It has become secondary parasite. Even in the after Secondary World War also, the soldiers were infected by this particular fungus and they had to lose their limbs also. So such kind of studies are very important, and G.P. Agarwal has contributed a lot. Late Professor G.P. Agarwal has contributed a lot for keratinophilic fungi. This is Chaetomium, which produces tons and tons of cellulase enzyme. This is Ceratocystis fimbriata. You can see bioluminescent astrocytes. Beetles carry the inoculum from one region to the other region. In the United States and other temperate countries, it is an avenue tree. All the avenue trees are infected by this wilt fungus. They are lost, and they could not trace out the way for the beetles, how they are carrying this. And mushrooms, you can see these are the Basidiomycetes, and nobody has asked to hide this particular net, Dictyophora, and then Polyporus. and Odemanciella, and the bioluminescent fungus. And you can see, next one please. The other fungi are these are the fungal biotechnology. As I told you earlier, the mushrooms are very important, and it is a multi-billion-dollar industry. Otherwise, in India, we have got a lot of more than 10,000 mushrooms, edible mushrooms are there, but nobody is there to identify them. Then food applications, bakery, and then brewing industry, cheese making, mushroom orientation, oriental fermentation, single cell protein. I told you, $16 billion worth of fungal products are available: alkaloids, antibiotics, ethanol, enzymes, gibberellins, immunomodulators, cyclosporin. It has revolutionized the organ transplantation. Without cyclosporin, it is very difficult. Organic acid, polysaccharides, paper industry, biological control, bioremediation. Dr. Jalali sir is an expert in the case of mycorrhiza and as well as in biological control, and as a plant pathologist may be interested. So this is how one should look into the group of fungi, and I will come to the end of this particular lecture. See, good science not only got monetary value, but also it has the power to purchase millions of health. That is very important. And coming to Professor Jalali's comments earlier, sir, when I was in DBT, I told Dr. Manju Sharma at that time that she has to give the projects. In the task force, I was there for 10 years. I have given the projects to many of the mycologists at that particular time, and told her that she should have a scientific eye for the mycologists and as well as for the mycological work. And recently also I was involved in the risk management and biosecurity, and with reference to plant pathogens and keratinic fungi, I have given my recommendation. They have accepted regarding this, and that is one. When I was in UGC for 15 years, I have given more than 1,000 projects for mycologists. And then I have the work that has been done, whatever the syllabus, the framing is there, I was a member, and then only algae and fungi were included in the first paper. Now they have merged with others. That is the tragedy. Now, later on in the DST also, I have created two centers, the culture centers, NCCC, National Cell Culture, and the other one is DST, Culture Center at Agarkar Research Institute. Sir, tell me, you are very kind to tell my mistakes to me, not to the others. I am very much thankful, sir. You have told me my mistakes only to me, not to the others. And last I would like to say is, दिखाई कम दिया करता है बुनियाद के पत्थर, जमी में जो दब गया है इमारत उन्हों पे कायम है. Thank you very much. I thank every one of you. I thank IPS, Dr. Pramila Devi, Dr. Rashmi Agarwal, Dr. Diva, and Dr. Gagoi, and all others who are here. Thanks to Professor Jalali sir for chairing the session, and Dr. Dubey and other rapporteurs.
Thank you very much. Profound thanks for a very brilliant lecture. And also summation with the spiritual thoughts, which can trigger the next generation of mycology.
Yes.
I was deliberately trying to trigger some debate so that people who are, in deep sleep might be possibly woken up and address the future challenges well in time. And I congratulate you with all your immense contributions you have made for the last more than half a century. And your efforts should continue to train the younger scientists.
Thank you, sir.
Also, when the molecular techniques have come into play. So it can be an ideal platform now to train the future generations of mycology. And we wish you best of health in the background of your post-COVID era. We wish you many, 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 many years to come to give mycology a great breathing space. Wishing you best of luck and success. Thank you very much, and God bless you. I hope the second speaker has already reported.
Yes, sir, she is there.
Dr. Shilpi Sharma?
Yes, I'm here. Dr. Jalali, I'm here.
Oh, good. Well, we have a second keynote lecture in this session. The second one is by Professor Shilpi Sharma. She's a professor in the IIT Delhi in the Department of Biochemical Engineering and Biotechnology. Briefly introducing the speaker, after having graduated and master's degree from Banaras Hindu University in the years 1997 and 1999, she has taken PhD degree from the Ludwig Maximilian University, Munich, Germany in 2003. She was inducted in the faculty of IIT and has worked in different capacities as a lecturer, assistant professor, as associate professor, and currently now she's a professor for the last, I think from November 2019. His contributions, she has made research publications around 58, 20 chapters, and also has made presentations at various national and international platforms, and has got many awards, including Innovative Young Scientist Award, and has also contributed a lot in the domain of PGPR and also on biocontrol concept. And also the award in terms of Inamdar Memorial Gold Medal, and also the Gayatri Prakash Dulari Medal, and so a long list of awards. A very accomplished scientist, and she has supervised 26 masters and 6 PhD students, and currently there are 9 students undergoing PhD program under her supervision. I invite Dr. Shilpi Sharma to talk on probing deeper into the biocontrol of fungal pathogens. Over to Dr. Sharma, please.
Okay, a very good afternoon, everybody. Thank you, Dr. Jalali, for the generous introduction. Sharing the same platform with stalwarts like Dr. Manohar Achari, Dr. Jalali. I mean, there's so many names. I have to say that it's definitely an honor for me. I would like to start with thanking Dr. Pramila and the other organizers for inviting me to share my bit in the area. Before I plunge into the topic, I have to admit that I'm not a mycologist, though my educational background has been in botany from Banaras Hindu University, where I was fortunate to have been trained by some great mycologists. I am an enthusiast of plant microbe interactions, primarily to enhance plant productivity in a sustainable manner. In this context, the triangle of plant, pathogen, and biocontrol agent excites me. Hence, I chose this topic for today's talk. So my prime objective in the last decade of my association at IIT Delhi has been to overcome the limitations of conventional agricultural amendments and to view them in the light of recent methodological developments, whether it is by employing omics tools or by devising newer strategies to manipulate the microbiome. These are some of the recent interests in my working group right now. I have been working towards assessing the non-target impacts of chemical pesticides as well as botanicals. By non-target, I'm referring to their impact on indigenous microbial community composition as well as on the function, so as to establish the disturbances and to estimate the resilience of the community towards such an amendment. Another area of recent interest is the surveillance of antibiotic-resistant bacteria and genes in arable land. Now, we're all aware of the increasing concern of the spread of ARB and ARG posing a global threat. The idea in this project is to assess the contribution of manure application in the spread and abundance of antibiotic-resistant bacteria and genes. Then I'm working towards understanding the mechanism of suppressiveness of plant pathogen by different farming practices. In fact, this is a project sponsored by a scheme which has recently been introduced to facilitate collaboration between IIT Delhi and ICR institutes. And I'm collaborating with Dr. Y.S. Shivai from the Division of Agronomy, IRI. Another focal area of mine has been mitigation of abiotic and biotic stresses in agriculture, especially because in the present scenario, we cannot afford to leave the stress fields aside, as India does not have a decent stand compared to many of the other nations in terms of arable land in hectares per capita. Under abiotic stresses, I've been working on amelioration of drought and salinity. This has primarily been by using plant probiotics like ACC deaminase producers, which have had a low profile till date. Under biotic stress, fusarium has been my favorite model system, and I've been trying to attempt its mitigation in different host systems. A lot of work has gone into gaining a mechanistic understanding of how this pathogen works, but we look at the problem from the perspective of biocontrol agents and its success in field. This will be the story for today's talk. The pressing need to shift to sustainable agriculture, especially imposed by the UN in the form of its SDGs, has brought back the focus on the practice of using environmental friendly alternatives for enhancement of both plant and soil health. Just to give you some numbers, some of you might already be aware of it. For a forecast period of 2019 to 2025, it is estimated that the biocontrol agents market is expected to increase from $3 billion US dollars, that was the market size in 2018, to $7.5 billion US dollars. And the countries considered in this study included not just some of the nations which have been aggressively moving towards shifting to organic farming, like some of the US and European nations, but also India, China, Japan, etc. Now, this is also because it's largely driven by the stringent regulations against the use of a range of chemicals in agro ecosystems. Hence, there's a need to revamp the technologies and the methodologies available for application of biocontrol agents. In the environment of a host, the pathogens, they coexist with biocontrol agents and other microbial members. The outcome of this various interactions between the three components of the disease triangle, that is the host, the pathogen, and the environment, determines the severity of the disease. In fact, it's been well established that the indigenous microbial community associated with the host serves in restricting the pathogens to a large extent. The mechanism of action of the biocontrol agents, whether present naturally in an environment or if they're introduced into it, they can be broadly classified into either direct modes or indirect modes. Now, we all know that the direct modes include, for example, production of antimicrobials, while the indirect modes would be more non-specific, like competition for nutrients and space, etc. Now, these mechanisms, they are not mutually exclusive. And the pathogen, in turn, has its own means to counter these strategies, like, for example, detoxification of the antimicrobials. So, in fact, in nature, you will find that there are a multitude of factors which determine the efficiency of a biocontrol agent. This ranges from abiotic factors like the soil physicochemical properties, the temperature, pH, moisture content, to biotic factors like the host properties, the associated microbial communities, all of which have a crucial role in the biocontrol agent's establishment as well as its efficacy. So understanding of the conducive environmental conditions for the biocontrol agents can help in the development of a highly efficient biocontrol agent. And such an understanding is all the more important with the recent concept of the plant being no longer considered as an individual entity. In fact, it's a meta-organism or a holobiont. That is the plant with its associated microorganisms or symbiont, with the latter contributing to its functionality. Hence, it's actually a zone wherein multiple domains come together, exerting influences from negative to neutral to positive. Now, having said all this, you can realize that these complexities impose certain limitations or challenges that have in fact impeded the application of biocontrol agents in fields. The performance of these microbes in field where multiple stresses act simultaneously is often inconsistent, and also it is quite reduced than that observed under lab conditions. Besides the struggles with the issues associated with a typical bioinoculant, like the shelf life, its slower action, in some cases even the economics. And as mentioned, the larger picture of mechanism of action of biocontrol agent is yet to be elucidated. The questions we tried to answer, and I'll only restrict to 2 of them for today's talk here. What is the larger picture of mechanism of action of biocontrol agents? Now, while a lot has been worked out, our emphasis was on assessing the active metabolites involved in this action and their impact in structuring the microbial community, the indigenous microbial community. And once we have this understanding, we wanted to know if it is possible to design some next generation formulations based on this information and knowledge gain, or which can overcome the limitation of reduced survival as well as efficiency of the biocontrol agents in the fields. Starting with the first question, the model systems were the ones well worked upon. I'm going to take you through 2 model systems on which we have worked currently. The first one, wherein the host we selected was one of the most popular vegetable crop grown across the world, that is tomato. And the pathogen in this case was Fusarium oxysporum. Now, we are all aware of the notorious effects of the fungus. The genus is also one of the most adaptive species. In fact, at places, losses of up to 80% have been reported with this pathogen causing vascular wilt. The management of this disease has been primarily based on mixed cropping practices, crop rotation, soil fumigation, using resistant cultivars, even application of chemical fungicides and biocontrol agents, biological agents. Various bacterial and fungal strains have been successfully used in controlling the disease. Genera like Bacillus, Pseudomonas, Trichoderma, Gliocladium, both individually and in some cases even there are reports of dual inoculations. For the present study, we selected a potent strain of Bacillus and another one of Trichoderma to first observe their combined effect in controlling the pathogen, as there are no reports of such a dual inoculation for controlling wilt in tomato. Now, we shortlisted these 2 strains based first after screening a collection of both indigenous strains as well as the strains that we isolated from plant rhizosphere as well as the established strains from culture collections. In fact, besides being positive for lytic enzymes like proteases and chitinases, these were actually multi-trait plant growth promoting strains. So, for example, the Trichoderma was an efficient IAA producer, while the Bacillus strain was positive for ammonia, for IAA, siderophore, even for HCN production. Now, Bacillus species are attractive candidates for use as biocontrol agents, also because we are all aware of it, because of their capacity to produce a wide array of metabolites. They're abundant in soil, so their establishment is relatively easier, and their ability to form endospores. The 2 together exhibited a synergistic impact on the plant with morphological change in this order. So what you see here on the left is the root system of an infected plant, while on the right is upon dual inoculation with Bacillus and Trichoderma in the presence of the pathogen. With a range of plant growth promoting attributes, even in the presence of the pathogen, the dual inoculation with Trichoderma and the Bacillus strains, they positively affected the plant parameters. In fact, the effect was even prominent at seed germination, which I've not included in the slide here. What I'm showing you here is the fold change compared to untreated controls at 3 different time points during the plant growth cycle. This was the best performing treatment with the 2 biocontrol agents in the presence of the pathogen. The maximum enhancement was observed at the first time point for most of the parameters. Nevertheless, even towards maturity, you will see a significant enhancement in the attributes. Now, with respect to the effect of the biocontrol agents in controlling the pathogen, it was found that the dual inoculation of B. subtilis and T. herzianum exhibited the highest disease reduction of 56%, even more than the fungicide, followed by their individual application. Now, here I'm showing you a comparison with carbendazim because it's commonly used against Fusarium, and also because it showed the best efficiency compared to 2 other chemical pesticides that we tested, mancozeb and chlorpyrifos. So having established that the dual inoculation was efficient in pathogen control as well as in plant growth promotion, we probed deeper into the mechanism of action of these agents. The question was, how does the introduction of this amendment structure the microbial community, especially in the rhizosphere, or how does the bacterial community respond to this introduction? When we compared the bacterial community profiles from the rhizosphere of the plants subjected to these treatments, something interesting came up. So while a distinct shift was observed from the controls wherein carbendazim was applied, the biocontrol agents, especially trichoderma, was seen to minimize the changes or let's say bringing the profiles closer to the control. We went on to identify the sensitive markers by sequencing the differential bands. Also, when we performed a qPCR analysis to get an estimate of the abundance of the total bacteria in these rhizosporic samples, the number were highest for the dual inoculation, as well as treatment with the pesticide, the fungicide carbendazim. And again, just like the qualitative picture, the effect of bacillus individually was subtle on the abundance of bacteria in the rhizosphere. So in fact, both qualitative and quantitative shifts were characterized post-application of these amendments. After assessing the bacterial community dynamics, the next obvious question was, what are the active metabolites responsible for the observed results of the application of amendment? So we performed an analysis of both volatiles and non-volatiles of the two biocontrol agents, both in the presence of the pathogen and without, using techniques like GCMS, LCMS, UPLCMS. The volatiles from the bacillus strains exhibited better inhibition of the pathogen's mycelium. Upon identification of the components, we found a number of unsaturated and saturated fatty acids, which have been reported to affect the membrane integrity of the plant pathogen. Now, for trichoderma, the most prominent compound that could be identified was in fact a terpene called santalol, which has been reported to have insecticidal role. Such a metabolite profiling was also done for non-volatiles of the two biocontrol agents for complete characterization. This was done using direct bioautography followed by UPLCMS. And in this analysis, the compounds like morpholine and various derivatives of piperidine could be detected. Piperidines are, as well as the triazine-based piperidines, have been reported to control phytopathogens like Pythium, Aspergillus, Candida species. So for this model system, we could identify the way in which the bacterial community is shaped by the amendment of the biocontrol agents and also the key metabolites involved in their action. So with this model system, currently we're working on designing formulations using different types of oilseed cakes. The practice of applying oilseed cakes, which are byproducts of the oil industry as organic amendments in arable land, it is usually performed in order to increase crop yield and also to enhance soil fertility. Hence, the idea was to use them not just for the protection of tomato crop, but also help in waste management together with enhancing soil health. And of course, this would help cut down the costs. We first checked the efficiency of 6 different oilseed cakes in inhibiting the pathogen's mycelium, and the results were most promising for the mustard seed cake. So we then attempted to use the cake as a growth substrate, both as cake broth as well as in solid state fermentation for the two biocontrol agents, Bacillus subtilis and T. harzianum. And we've also extensively characterized the mustard seed cake to identify the metabolites responsible for its antimicrobial activity. We have established the efficiency of such a formulation in pot experiment, and we are yet to perform the field experiments with this model system. So now with all this analysis of the specific metabolites which contribute to the antimicrobial function, we wanted to bring such a knowledge to application. For this, I'll switch to another model system. The host this time was pigeon pea and the pathogen Fusarium udum, which again is a soil-borne pathogen, highly specific for pigeon pea crop. The pathogen not only results in yield decrement, but it also adversely affects nodule formation in pigeon pea crop. So the choice of biocontrol agent in this case was actually a rhizobial strain, Bridyrhizobium, such that we could tackle the issue of controlling the pathogen as well as limited nodulation simultaneously. Now, Bradyrhizobia, besides the ability to fix nitrogen symbiotically, also improves plant growth by siderophore production, by phosphate solubilization, indole acetic acid production, secretion of various enzymes. While their plant growth promoting properties have been studied extensively, there are limited reports available on their biocontrol activity against fungal pathogens. So with this model system, the question we wanted to address was if the understanding of the bioactive metabolites of biocontrol agents can help in designing next generation formulations with enhanced survival and efficiency. The conventional bioformulation is based on using cells in a matrix. We compared such formulations with cell-free formulations based on pure metabolites and also cell-free culture supernatant, CFCs, which was a mixture of metabolites. So for metabolite-based formulation, we purified the EPS secreted by the bradyrhizobium strain, as it was shown in some of our earlier studies to have a role in establishing the strain in the plant's rhizosphere. The reason for designing bioformulation with CFCS was the first set of experiments which showed its inhibitory potential against the pathogen Fusarium. We set up plant growth experiments with several treatments. So we included treatments of just the rhizobial cells, the supernatant, and EPS individually, as well as their combinations. First, the effects were observed under controlled conditions in a plant growth chamber. The left side of the bars correspond to treatments without the pathogen, while the right side includes the pathogen as well as various combinations that I just mentioned. The triple combination of the rhizobial cell, EPS, and the supernatant led to over 2-fold enhancement in root length, and in fact, more than 1.5-fold enhancement in shoot length and dry weight over the control. The same treatment performed best even in the presence of pathogen, with approximately a fold enhancement in these parameters. Similar trend was also observed with nodule number. If you see, this is the triple inoculation with the pathogen and without it. Though here the enhancement in the presence of the pathogen was lesser compared to the corresponding treatment without the pathogen. And the triple combination additionally possessed the ability to suppress the disease with lower disease incidence compared to the other treatments. So the combination of the rhizobial cells, its EPS, and the supernatant was in fact exhibiting a tripartite cooperation. It was serving as a nodulation enhancer, as a plant growth stimulator, as well as a disease suppressor. For validation, we took this to the field conditions to monitor the plants for the entire growth cycle. And the trend in this case also remained the same, with the best performing treatment to be the triple combination. We then went on to characterize the cell-free culture supernatant. It had shown inhibitory activity against the pathogen in plate assay as well as in experiments with the host. The metabolite profiling of the cell-free culture supernatant revealed the presence of prominent compounds like derivatives of benzoic acid. Now, antifungal activity of benzoic acid and its derivatives has been established for various fungal phytopathogens like Aspergillus niger, for Phytophthora capsica, Rhizoctonia solani, and so on and so forth. The derivative of benzoic acid and salicylic acid have been reported to possess antimicrobial activity against a range of pathogens, whether they are bacterial, viral, or even fungal pathogens. The presence of amino acids like phenylalanine isoleucine in the supernatant, this might have a significant role in plant health promotion, including effective symbiosis between the legumes and the rhizobia, the positive impact that I just showed you. In fact, phenylalanine is also related to the synthesis of condensed tannins, which suppress diseases. Coumarin is a potent natural pesticide that inhibits fungal and bacterial pathogens, causing seed-borne diseases. So with this model system, we could not just develop a novel formulation with multiple benefits for the crop, we could also get a deeper understanding of the mechanistics of the biocontrol agent. Fine, so what is it that I can summarize with the two model systems that I talk about? We saw with the first model system that the changes caused by the fungicide to the bacterial community was to an extent larger than that exerted by the biocontrol agent trichoderma. And in fact, as a biocontrol agent, trichoderma also neutralized the impact exerted by the pathogen. We can therefore tag it as ecologically safe in terms of its impact on the resident microflora. With the second model system, we saw that when metabolites were used together with rhizobial strains, it not only enhanced nodulation, but also disease suppression and promoted plant growth. This actually paves way for the option of mining and purifying metabolites, and then probably directly using them for street drenching, for enhancing grain yield and mitigating the constraints associated with biotic stress of the desired crop plant. Additionally, it can also be adopted as a strategy to enhance the efficiency of known formulations. Now, what we are now working on is to design formulations with other metabolites that have the potential to control pathogens. Then we're also in the process of validating these experiments in farmers' fields. And another aspect that I'm interested in is to develop high throughput method for quantifying these metabolites so that they can serve as markers of soil suppressiveness. With that, I would first like to acknowledge the members of my working group who contributed to the stories that I talked about. This is Dr. Sakshi Tiwari and Dr. Monica Jangir. In the work, my collaborators were Professor Satyavati Sharma from the Center for Rural Development and Technology at IIT Delhi, and Dr. Anil Saxena has been quite pivotal in providing us all these trains. The generous funding was from SERB and from DBT. And I would like to congratulate the organizers for this excellent platform despite the current situation. Last but not the least, thank you all very much. Dr. Jalali, over to you.
Well, thank you very much. Are you able to listen to me?
Yes, Dr. Jalali.
Okay. Thank you very much, Dr. Shilpi Sharma, for very interesting presentation of good work done by you and your group. In fact, biological control has been one of the very active areas of research all over the country at various platforms, and your work in that dimension has been quite useful. At the same time, often a debate is being raised at various platforms with regard to the quantum of work which has been carried out at the national level has been really remarkable. Good quality research has been done, which has been documented in high glossy journals and so on and so forth. Nevertheless, this in vitro information which gets generated somehow does not get translated in vivo. Therefore, my only submission with all the active biocontrol agent specialists would be that we need to walk an extra mile to see how far these in vitro studies get matched with the in vivo, or what you have at one point of time you have written you want to validate at farmer field. In fact, this is a note which I have been seeing from all biocontrol agent specialists all over the country, this statement being made. My question therefore would be, we should straightway headlong go now, but wherever we are finding very positive results, in your case also very nice information has emanated. But now, because any recommendation which has to be made has to be not only scientifically sound, it has to be economically viable and also socially acceptable. Then on, in fact, that reminds me of a great microbiologist who said, to him who devotes his life to science, nothing can give more happiness than making discoveries. But the cup of joy is full only when the results of a research study find practical application. So Shilpi, this information which has been very nicely done by you and your group, kindly make serious efforts now to get translated so that important recommendations can be made at the national level. Biocontrol agents, you mentioned trichoderma, and a lot of work, as you must be fully aware than me. But then the quantum of information suffering from plants, because trichoderma
Dr. Jalali, I'm losing you, I think. Is it the same issue with others?
Yeah,
Yes,
it
his
may
voice
be
is
poor
not
net
audible.
I
connection.
think it is cracking some. Anyway, so I would just like to add to what Dr. Jalali said. I mean, it is. I think he pointed out exactly what has been going on in our group and the philosophy behind it. So it has to come to the fields. And most of the work that I have shown you was not restricted to in vitro. We validated everything in the experimental fields, but I did not want to, unless we actually show it in the farmers field. In fact, IRI has been pivotal in bringing all my research to translation. But unless I show it in the farmers fields, like Dr. Jalali said, it has to be shown there. There's absolutely no doubt about it. And as I speak, I have experiments set in farmers fields in Kanpur Dehat, where there is a lot of issue with the pathogen. But I would not like to, such preliminary data is definitely not going to make up a part of my slide for a symposium of this sort. So probably next time I'll be able to even show you the results from farmers fields. But most of the results I showed you was from IRI fields and the experimental fields at IIT Delhi.
Very well done, and very well. Wish you best of luck and success.
Thank you very much. Thanks a lot.
I see Professor Niranjana has joined somewhere, right?
Yes, I also see him.
Hey, where are you, Niranjana?
Unmute करिए sir, unmute.
I am here, sir. Good afternoon, all of you, sir.
जरा कहाँ नाश्ता किया है? जरा नाश्ता.
Good afternoon.
Good afternoon. Good afternoon to everybody.
You don't seem to have taken breakfast.
No.
Hungry at lunch.
Even otherwise, also I am strong.
Okay. Now, any question to Dr. Shilpi?
Not the question. I have some clarifications.
Where is she?
Please, sure. I'm here. It's just that I'm about to go for another meeting, but please, I'll be happy to answer. Yeah,
okay.
Wanted to make two clarifications, very simple. Is there any assay to determine the activity of effect either with the plant growth promoters or the back to compound metabolites?
Can you please repeat again? I think
to determine whether the effect of biocontrol agent either from the plant growth parameters or from the bioactive compounds.
Okay. So you want to say if we can separate it out. What we have done right now is we have tried to generate some mutants wherein these pivotal properties of plant growth promotion have been compromised. And we are now trying to see if these metabolites are actually helping. And also we have done certain experiments wherein we have even used the pure metabolites just to be sure that it's the effect of the metabolite which is resulting into those observations. These have been done. Yeah, we can differentiate that.
We can differentiate.
Yeah.
My second clarification is that have you studied any shelf life in oil seed cakes?
Yes. So in fact, this was the first experiment with oil seed cakes that we did, and at a constant temperature, we could see that the shelf life was as per the norm. We could have a decent shelf life till about 6 months. But we are definitely working on even some. The reason for using oil seed cake was because it was actually offering a very protective environment to the inoculants, the agents. But we are also trying to now enhance that. So, but this is underway. So I will not be able to give any data as of now. But yes, this was the first experiment we did to see if the shelf life was being enhanced.
That's good. But see, normal culture will give the 6 months shelf life.
Yeah.
So when you add some oil seed cake or your back to compound is specific, so it must have some more time of shelf life.
Correct. But sir, it's also important that you have a fluctuating temperature, and not just for Indian scenario, we should also have that. So this is the reason why I said that we need to check even under fluctuating temperatures and not just one constant temperature.
Because the last clarification with me, Professor Jalali, with due respect, he said that in vitro, in vivo, and we are has the success with this bioactive compound now, not with the culture, and we are going out with the small medicine of a bioactive compound in a vial and success in the field condition to spray for the targets. Thank you very much.
Okay. Congratulations, Dr. Shilpi, for very nice, interesting piece of information.
Thank you very much, Dr. Jalali.
And wish you best of luck and success in your future endeavors.
Thank you so much. Thanks a lot.
Well, friends, now these were the two key speakers and very fascinating lectures. and we are enlightened by both the speakers. Wealth of information has come forward, and I wish, therefore, I would like to thank both the speakers and my co-chairman, Dr. S.C. Dubey, and reporter, Dr. Vishnu Maya Bashyal, and who is Pradeep Kumar kahan hai? I don't see him around here.
Here, sir. I am here, sir.
Pradeep,
बहाने आपके दर्शन तो करूं.
Pradeep Kumar. Pradeep Kumar.
Pradeep Kumar. Pradeep. He was supposed to be one of the
Reporters.
I am here, sir.
Now he is here. So I hope you must have taken note of all this, what has been said.
Yes, sir.
Vishnu.
Yes, sir. Yes, sir.
Take care.
Yes, sir.
And I wish to thank the organizers, Pramila and her group, for giving me an opportunity to share some of my inner vibrations with you all. Thank you very much, and God bless you all.
Thank you, sir.
Thank you. Thank
Thank
you,
you
sir.
for
Now-
accepting the invitation.
Thank
yes,
you, sir.
Amrita.
So now it's time for the lunch break, and we'll resume here at 2:30 for the next technical session. Participants for the poster session should continue with the same link, and participants for the academic merit award, they have to join another link given in the technical program.
There is no online lunch available.
Everybody can have on their seats, no?
Online
for contest na academic award.
Send the bill, sir, online. Send us the bill.
No. That is not the right.
You get from there.
Basically,
Sir, you come once, we'll go for lunch. That's a better option.
Yes.
Anyway.
You tell him now. No, you tell him lunch can be sent through WhatsApp.
Lunch
can be sent through WhatsApp.
2:30. 2:30. Yeah. 2:30. It is announced.
Have a good lunch. All the best.
Hello.
Shall we?
I think we can now.
Yeah,
We can leave now.
Sir, no need to leave this link. We can.
Link will be there. Yeah.
And another people who want to join the award lecture, they have to join with another link. This will be continued. I'm not closing this link.
Don't close, but let it be there.
Yeah,
Right.
it will
After
be like
lunch,
that only.
everybody will join.
Yeah, everybody come for the poster session.
So we all will join at 2:30.